Okay, so welcome back, everyone. So now we will continue with applications of using different types of descriptors, modeling methods, and so on in different areas, chemicals, pharma, consumer goods, and so on. And so Jennifer will tell us about the use of bioactivity descriptors for in vivo tox prediction. Jennifer, feel free to go ahead.
Thank you. I guess we've heard a lot today already about molecular initiating events, about different pathways where toxicities can occur. And so my talk, I think, is hoping to combine a little bit the topics of similarity of different descriptors of machine learning. So what I'm going to tell you about today is about how we can use different notions of molecular similarity to help our machine learning models to predict in vivo toxicity a little bit better. So I want to start with the definition of chemical similarity. So if we talk about structure-activity relationships or even quantitative structure-activity relationships, we basically assume the principle that if we have a similar structure, we assume that they also have similar activities and therefore, of course, also toxicities. But if we look at those two examples that I want to show you, is if we just very slightly change a structure, so we go from a ring system and then we put these amine group there, we go from a non-mutagenic chemical that we want to have, that is what we desire, to a chemical that is mutagenic in the Ames assay. So here we have a minor change in the structure and we have like a, let's say, large change or an opposite change in the activity. And on the other hand, if you look into other compounds, it gets even more, let's say, subtle where we have the differences. So here we go just like from the R configuration of the molecule, we have a known sedative effect that we want to have and that we desire in this pharmaceutical. Whereas if we go to the S enantiomer, we have a teratogenic effect. And so I think like if we go back to this structure similarity principle, is that we need to a little bit rethink how do we show these molecules to our machine learning system so that this machine learning system is also able to know or to learn these subtle differences. And so I think here on the left-hand side, so if we talk about structure similarity, what we usually talk about is fingerprints, somewhere descriptors, so be it physicochemical descriptors or other calculated values from a different structure, but also nowadays more learned representations, so graph-based fingerprints, normal encoder-based fingerprints, and so on. But something that most of these representations have in common is that they might be able to trace these subtle differences, but usually they are not able to trace a three-dimensional configuration changes. However, and that's something that I guess we have learned today, is that we have these types of molecular initiating events where we, for example, have biological assays to really see these differences in molecules. So even though we say they might be chemically very similar in terms of the representations that we can use for machine learning, we know that those are biologically different. And so why not tell our model more instead of only about the structure, but also about the biology that we are looking at. And here, I think in the last years, there have been large leaps. And if we look towards high throughput screening data, for example, the ToxCast data set, I think was a huge success. We can also have dedicated assay data. For example, for the Ames assay that I mentioned before, we have large assay data in the public domain. So if you look towards carcinogenicity, I guess the Ames data might be a really nice proxy when it comes to biological behavior. But we also have heard today also about omics data that we can use, where we also see very differential bioactivities pretty early, even before we can really realize something or look at the cell and see the cell is going into apoptosis or is dying in our cell culture. And then, of course, also something where we had the session on is also PBPK or PBTK data, where we know that, for example, a compound only can exhibit toxicology or toxicological phenomena if it's, of course, if it's taken up into the body. So when we thought about this, so what we came up with is what we call bioactivity descriptors. And however, so there are large data sets there. We have Also internally, we have a lot of compounds that we test in different assays. However, a problem that always occurs is if we look into data sets, we nearly never have a data set where we have a complete matrix of compounds that were tested in all assays. So even we look towards the ToxCast assay, even there, not all chemicals were always tested in all assays. So you always have these data gaps. And now, of course, the question is, what do we do with those? And of course, I guess the most easy thing is to go and say, well, we're just going to measure all those values. For high throughput screening, that might work. But if you look more towards specialized assays or PBPK, that is, of course, something that is super hard to achieve. So here we need a synthesized compound. So we even like if you look towards animal data, of course, a lot of synthesized compound. It's quite expensive. It's time consuming and probably it won't really help us to getting our results faster. However, of course, if we measure all these values, we always have like a fixed assay in the lab because we can do that in a specific lab at the university, internally at BASF. And of course, if we know the assay and the lab, we also have a known uncertainty. So we know how good the assay is. However, of course, I mean, measuring is not really feasible. So we thought, why not predict those missing values? And here, I mean, what we can do is we can predict those values for compounds that we do not even need to synthesize. It's pretty fast and expensive to do in silico predictions, but of course, and I guess that's what we're all struggling with, is we need standardized data to achieve this, to train good models to then be used as a proxy for measurements. And there, of course, I mean, the model quality is very crucial. And something that I want to pick up later is that, of course, if we have an assay with a defined applicability domain, the question is this like applicable to novel chemistry? So over the years, if we're developing compounds further, if new scaffolds get into trend, the question is like, can we use this in the future as well? So what did we do for our project? So we called this project ChemBioSim. So and we thought that if we can use in vitro assays to predict in vivo activities, we can really define hazards early on. And I think here this concept of using in vitro assay data as a proxy for in vivo outcomes is something that we also have like heard of today, and that we can maybe also put a little bit into the scheme of molecular initiating events, because what we measure in vitro is usually, for example, toxicity to specific cell types, the Ames assay, pretty easy endpoints that we then want to use as a proxy to predict in vivo activities. And so what we try to do is, I'm sorry, my docking station. Can you still see my screen?
Yeah, the screen is fine.
Okay, perfect. So what we try to do is then, we thought that why not use in vitro assay data as descriptors for machine learning. So we started with having like a labeled bioactivity data set, just calculating normal structural descriptors, for example, fingerprints, and in the RD kit, the physicochemical descriptors, then divided this in a training and a test set, used the training set to come up with conformal prediction models for the bioactivity values, then of course evaluated the test set, and then we basically plugged those bioactivities descriptors in for the in vivo data set. So all the in vivo data was predicted with our models for the bioactivity descriptors. Then we joined them onto structural descriptors, so here fingerprints and RD-kit descriptors. And then again, we went into splitting into training and test set, trained our machine learning models for the in vivo endpoint, and then evaluated the data. So what types of data did we use? So we started with the available data that we found in the literature. So here a few, let's say, single endpoints like the Ames, thyroid, KCO2. And then we were lucky enough that groups published two larger databases. So the EMOLTox data set by G and colleagues with 136 endpoints that we used. And the ToxCast database that I mentioned earlier, which was published, I guess, a few years back by the EPA. So here we also had 222 endpoints. So we had with roughly 350 endpoints for the in vitro assays. And what we made sure of is that each data set had at least 200 actives and inactives. So we had a reasonable size of training data, and also, let's say, a reasonable amount of actives and inactives where we could learn from. For the in vivo data, what we used was we compiled 3 data sets. So one for the micronucleus test, which also indicates if a molecule is able to damage the chromosomes. A data set for drug-induced liver injury, which we also already have heard about today, and a data set for drug-induced cardiological complications. So what you can see is here that the drug-induced cardiological complications data set is the largest data that we had with roughly 3,000 compounds, and that the DIG data set and the MNT data set were both fairly unbalanced. Whereas here for the DILI data set, it's a fairly small data set, and it was not as imbalanced as the other 2. So what did we do? So in our first case study, we of course wanted to see what can we achieve with these bioactivity descriptors. So are they essentially useful to use, and are they essentially helping the model to really learn different endpoints better? So what we already did, and I briefly mentioned that previously, is so we took these structural descriptors here, very specifically the Morgan fingerprints with 2,000 bits, the RDKit descriptors, so roughly 2,170 descriptors, joined them onto the bioactivity-based descriptors for all the endpoints that we trained, and used there. We didn't use the final classification, but the p-values that were calculated from the conformal prediction model for both classes. We joined them together, and then what we first did is we of course did a feature selection, because those are way too many features to put into a machine learning model, and then trained a random forest, a conformal predictor on the in vivo assays. So I guess the nice thing is, and I probably wouldn't tell you about this if it wouldn't work. So we really saw that this can work, and this can really improve the efficiency and the accuracy of the models. So here, maybe for those that haven't really heard about conformal predictions, the efficiency is basically the amount of compounds that were predicted with a single class label. And this means that those compounds were predicted as in domain, and what we saw here is that the amount of those compounds were really we could be increased for the micronucleus assay and for the cardiological complications, but it didn't work for the liver injury. And I think in concordance with this, if you look into the F1 score, which is basically the harmonic mean of precision and recall, so basically a measure of how good the actives can be retrieved, is if we look here into orange, the bioactivity descriptors, we were really successful for the micronucleus, and we were really successful when it comes to the cardiological complications. However, so overall for the liver injury, it did not work as well as for the other 2 endpoints. But I think something that has to be put in favor of the model here is that here you saw the data set comprises of roughly 600 to 700 compounds. And this is a super limited amount of compounds for such a very complicated endpoint such as liver injury. So we haven't looked into it fully, but we hypothesized that probably it's due to the quite low amount of data compared to the very complex endpoint, that here we couldn't really achieve a much better performance. And of course, also we see that here we are in the ranges for the accuracy of roughly, let's say 75 to 80%. It could also be that here the model, that the baseline model is already at the, let's say, at the threshold of what the data gives us. So we probably also are in an area where we might not get much better than this, irrespective of the model or data that we use. So now we've tried this with a random forest, and it worked very well. Now, of course, the more interesting thing, also if you look into the liver injury, is can we maybe use a more sophisticated algorithm for the later prediction model? So what we kept is the structural descriptors. We kept the bioactivity descriptors trained on the random forest, but then we changed the ultimate model where we input those features into different algorithms. So we used a random forest, we used a support vector machine. We wanted to see whether like XGBoost has like a performance boost over the normal random forest. And then we used a graph-based neural network called ChemProp, which was developed by the MIT. So now the interesting thing is maybe can more sophisticated algorithms, like for example, a graph-based neural network, really achieve a better performance than, let's say, a baseline random forest. And so here to guide you a little bit, so again, in green, we have the bars where we didn't use the bioactivity descriptor, orange, the bars with the bioactivity descriptor, again, the micronucleus assay, the liver injury, and the cardiological complications. So I think overall, what we can see is the same pattern than for the random forest. So for the micronucleus assay here down here, it's the F1 score. For most of the models, we could improve the prediction with the bioactivity descriptors for MNT, and especially here for the cardiological complications. The same goes for the efficiency. For the cardiological complications, it was more pronounced than for the MNT. Again, for the liver injury, we unfortunately didn't see any improvement of the bioactivity descriptor. And so overall, the pattern was the same, but unfortunately, what we could see is that we don't really see a large improvement depending on the algorithm that we use. So here, what you can see is that the forest-based methods seem to perform a little bit better than the support vector machine and the ChemProp model. But overall, I would say that most of these differences are not significant. And so the predicted performance is more or less in the range where we would expect it, and where we also, I guess, was assuming that we cannot really improve the performance. And again, so I was hoping that maybe the ChemProp model, so the neural network, was a little bit more performant when it comes to liver injury. So you see it's in the range of the other models, so there's no performance boost. Again, maybe with a little bit larger data sets for the liver injury. However, with this small problem that if we look here into the cardiological complications, this is a larger data set than liver injury, and even here we don't see a large boost of using a fancy neural network over the standard random forests. So nevertheless, I think that was super interesting to see, and I really was satisfied and content that now we see these boosts not only for the random forest, but also for other machine learning algorithms. But now, if you remember that I said at the beginning that the applicability domain is something that we are concerned with, is now I want to go a little bit away from the sole bioactivity descriptors, but into the idea that if we have like in vitro data that we are using to predict in vivo endpoints, and if we then go into the scenario that we are only predicting these in vitro assays and not conducting them anymore, that of course over the years, the chemical space that we trained the model on and that we are predicting the new compounds that we are developing over time at BASF, is that those spaces are probably differing, and that those spaces are probably diverging. And this is what I really want to tell you in the third case study. And so if we are changing the chemistry over time, so can we still apply our models? And now, if you had never heard about a conformal prediction, I want to give you a very, very short primer, but I'm not going to go into all the details. So the difference between conformal prediction and normal prediction models is that actually we do not only have like a training set, but during training we use a calibration set. And this calibration set helps us to basically to rank our predictions and to calculate something that is called a non-conformity score. And this score is then later also used to calculate the p-values that I was earlier mentioning. So these p-values are not the statistical significance p-values that you know from various papers, but very, let's say, conformal prediction specific. And so now the interesting thing is that this calibration set can really help us also, for example, if we have imbalance data, to really help us to calibrate our model to better predict the imbalanced data sets with regard to the positives or the negatives, depending which is the minority class. And so the idea was, well, basically, if this calibration data set is what helps us really to also be able to tweak our model towards predicting the minority class better, like is it enough to only change the calibration data set over time? So that means that we just need to measure, like, I don't know, once a year, a few compounds, we exchange this calibration data set, and then we can really use our model trained on the original data set and use it on our novel compounds. So what we did here is we used Campbell based on different assays, and then based on the dates where the assays were conducted. So we had the full Campbell data set, and we had the older data and newer data, and then we started splitting. So we had the old data kind of for training and calibration. Then we had 2 newer calibration data sets, and then the newest data was used for holdout as a test set, which we were using then to basically rank whether the calibration updates were really working. So what did we see? So we start here with the validity, and the validity is something for conformal prediction, which is super important because the validity guarantees us that our model is essentially can be used on the data that we are using. So we started, so we set the threshold at 80% for the validity. And so in the original training, we see that it definitely worked. So here we had a validity of 80%. Now, if we use our holdout test set, what actually happened is that if we use the holdout test set on the original calibration and the original training data, the validity drops. And this just means that our assumption that 80% of the predictions that we get out of the model are correct is just not true anymore. So that essentially means that we cannot use the model on this data because we cannot assume that the predictions are valid. So then we used our calibration data set 1, the calibration data set 2, and also like we try to combine these calibration data sets just to have more data and to see if maybe more data for the calibration gives us an asset. So what we could see is that we could restore the validity, so we could really use the model on the holdout data set. However, what unfortunately happened while we tried to restore the validity is that whereas we started with a very good balanced accuracy, and here on the right with a very good efficiency on the training data, that the balanced accuracy as well as the efficiency unfortunately dropped while trying to recalibrate the model. And so what we see basically is the balanced accuracy dropped, which is something that you might expect because the model was trained on data that was not, that is basically from a different chemical space than the holdout test set. So we kind of assume that the balanced accuracy is dropping. And similarly, so what we see here is that the efficiency is dropping. That means that the model is starting to predict more compounds as out of class compounds, which kind of is in combination with the balanced accuracy. So although we could restore the validity of the model, so we can say we are allowed to use the model on this holdout test set, unfortunately, although let's say that we again have the statistical guarantee that it works, we see that the model is struggling to predict these out of domain data, also with the recalibration, because the balance accuracy drops, and with this the efficiency. But I guess it's also pretty interesting to see that the model here starts to recognize much better what is out of domain and what not. But unfortunately, to say here is that we can restore the validity, but we cannot really only recalibrate the models. But if we see that the chemical space is changing, and if we keep track of our validity for future predictions, we really need to retrain the model based on the novel data that we have. And so I think with this, I want to conclude. So I think we saw that bioactivity descriptors do have an impact on the predictive performance of machine learning models. And of course, now seeing that it doesn't really have an impact on the in vivo models, which algorithm we choose. Nevertheless, I would like to see what the impact is on the algorithm choice for the bioactivity descriptor generation. And yeah, we unfortunately didn't see like a lot of difference on the different algorithms, but I think for all models, we had a pretty high baseline predictivity. And maybe in the future with novel data, so we can also see maybe benefits there, depending on I guess, how hard the data is to predict. And then at the end, I could show you that if we have data drifts, we can really mitigate them by recalibration and restoring the validity. Unfortunately, that doesn't like change the applicability domain of the model. So although we had a new validity, we couldn't really adjust the applicability domain. So that means that we really need to retrain models in the future, also if our chemical space changes. And with this, I guess, like I didn't do most of the work, so I really have to thank Marina, who was our PhD student, who did most of the work, Miriam, her supervisor, and many other supervisors from other universities and institutes. And with this, thanks for your attention.
Excellent, great. Thanks a lot, Jennifer. Is there, we have time for one question, I think. Yeah, regarding sharing the presentation, by the way, I've received 5 or 6 slide decks already. Feel free to send them to me as well, Jennifer and all the other presenters, and I put them on the website when I receive them as well. Okay, let's do one question. Martin?
Thanks very much. Sorry, just finding the mute button. Thanks for the talk, Jennifer. I was just interested in the first part where you're talking about the bioactivity descriptors. You showed them compared to the chemical sort of fingerprints, and then you talked about feature selection. I just wondered if you could say anything after feature selection. Was there anything you could learn from which bioactivity descriptors remained and whether some were more predictive for particular in vivo outcomes?
Yeah. So what we actually could see, also looking at the like feature importances that we also did, is that sometimes we could really find good correlations. So for example, the Ames assay was very important in the prediction of the MNT data. For the drug-induced liver injury, also found a few correlations that were like, for example, that were very, let's say, reasonable. We, of course, also found correlations with, let's say, new in vitro endpoints where we couldn't find any literature, whether those were really relevant or whether there is a link, which either might be a link that's not seen or just some spurious correlation. So sometimes it was helpful, sometimes we couldn't really establish the reason why this is. But most of the times, really, we saw that bioactivity, so after feature selection, we had quite a few bioactivity descriptors there. And we also saw that, of course, the models for the bioactivity descriptors that were better were more often selected than the ones that had, let's say, F1 scores, which were lower than 0.5. So that was also interesting to see that actually there seems to be, the model seems to be selecting based also on the predictivity of the previous model.
Okay, great. Thanks. Thanks for the answer. I think there's one more question in the chat, actually, and I think there was one hand up. I would suggest, because we are out of time for this presentation, maybe, Jennifer, you can follow up in the chat with the other questions. Great.
I'll do that.
Excellent. Thank you for your contribution. And we'll move on to Pharma, and David is presenting next.
Yeah, thank you very much.
Yeah, if you stop sharing, that would be great. And then
I'll try to figure out where that is. I'm sorry.
Yeah, I think I can do that here as well. Let's see, stop sharing. Yeah.
Okay, then, thanks.
Excellent. And David, I think you can share slides and then.
Oh, can you see my slides?
I think it's coming. I see. Yep, absolutely. All good.
Perfect. Thank you. So thank you very much, Andrea, who actually organized this meeting, and thank you for your attention for the online audience of this talk. My name is David. I come from Roche in Basel, and today, if I summarize the presentation in one word and in a 2-character word, would be hi. It's a talk that we prepare as a team that we call the computational toxicology, and we want to say hi to the community and share what we are now doing. And the title of today's talk is Traverse Causal Modeling of Drug-Induced Toxicity for Preclinical to Clinical Translation. And this is actually where our team see ourselves is going. So we are a very recent team, so we just started this year, though Roche has a long tradition as any other company working in the field of computational toxicology. But it's just this year we start to have a dedicated team on computational toxicology and safety. And we got broad input from Roche colleagues as well as external experts. To start, I want to share what actually other colleagues already said at the beginning of today's meeting, also in the last meeting, very nicely illustrated by Jennifer. We face a lot of challenges in preclinical safety assessment and risk mitigation, starting from the ever higher safety hurdles and scrutiny by both general public, but also by regulatory agencies. We are now embracing more and more new modalities, from gene therapy to also cellular-based therapy. And we are, as always, committed to replace and reduce animal use. And this is not always coming with easy tasks to predict what happens in human. And we are facing evolving R&D models for those who do not work in pharma. Classically, research and development is very close integrated, but now very much the research is done outside the company, and only near the clinical phase, the asset is brought in, and very often with very limited or scarce information about the safety profile of the drug. And last but not least, I think in every industry, there is always a time and cost constraints. So these are the challenges we are facing. So that's why we actually decide to set up this computational toxicology and safety group, because we are convinced that when there are challenges, there will be more solutions. And on the positive side, we have more data awareness, availability, and fairness. This has been touched since the beginning of today's meeting, which I'm very glad to look and to observe. We have, as other speakers talk about, high throughput platforms with high dimensional data. We have newer human model systems, from simplistic co-culture model to complicated like spheroid or even organ-on-a-chip type of models. We have off-the-shelf in silico models, as well as tools to create bespoke ones. We have diverse minds of working on translational safety. So in our group, we have both computational biologists, toxicologists, also the first author of the last work presented by Jennifer, Andrea Morgan is in our team. So we have very different mindsets working on translational safety. And last but not least, Roche has made the commitment to do now what patient is next. So I said we are a new team focusing on computational toxicology in preclinical to clinical translation. We query which issues in drug benefit risk assessment and risk mitigation can be solved by data and computational methods, and address these problems as a team in collaboration with other experts, both internal and external. So our work is mainly in three areas. One is to discover new data sources for toxicity assessment and prediction. This may include human model systems, clinical data, and real-world data. We identify relevant feature types. This can include single cell and spatial omics, PK/PD data, as well as molecular modeling and simulation. And last but not least, we develop bespoke models fitting the question. This can include generative model, interpretable machine learning model, and causal inference. Our vision is to perform causal modeling of drug-induced toxicity for preclinical to clinical translation. So our team now work on three main focus areas. One is to building computational models and co-developing new in vitro assays to screen and profile preclinical drug candidates for safety liability. And this I will show now with one case study. The second area is to improve our understanding of molecular and cellular mechanisms causing toxicity and suggest possible mitigations. And this we have talked a lot today, from the initial molecular event, also the other aspect of the mechanism, AOPs, and we are actually working on that aspect as well. And the third is to establishing the causal chain between molecular mechanisms, exposure, patient characteristics, and real-world observations of efficacy and safety. And there we rely on both machine learning type of models as well as causal inference models. So I said this is now a hello presentation, a hi presentation, and therefore I chose only one example that is a recent piece of work of our team. work on the optimization of the teratox assay for preclinical teratogenicity assessment to give you a flavor what we are working on. So the problem is actually also very nicely shown by Jennifer in her slides, teratogenicity of, for example, thalidomide. If you ask people outside of pharma what scandals do they know about pharma, I think one of the top answers would be the teratogenicity of thalidomide in the 1950s. And it's almost another scandal, but much less known, that after 70 years of the scandal, we still do not have a human relevant system. The most relevant system for this now is actually a mouse EST as well as a zebrafish model. So the current standard for preclinical teratogenicity assessment uses mouse cells and animal studies, which do not work well for human prediction. And that actually the problem drove us to develop a new assay, which we call Teratox, with a new readout molecular phenotyping that empowers predictive modeling and mechanistic understanding. So what we did, we performed expression-based analysis of 51 compounds with curated teratogenicity labels in dose response, and these human pluripotent stem cells. And using a platform that we built within Roche called molecular phenotyping, we could apply feature selection method and also use a statistical method called factor analysis, which actually we found that is not only in this context a statistical method, it reviewed actually biological insight about germ layers and stemness. Using this data, we could build a machine learning model that predicts teratogenicity. So we used factor analysis, random forest, elastic net, and Bayesian network for our approach. How the assay is set up? We use pluripotent stem cells, culture them for 7 days, and these cells go into spontaneous differentiation. They form this kind of very nice structure known as embryonic bodies. And these embryonic bodies, they are on one side tested for cell viability. The other side, they are subject to gene expression analysis, from which we derive germ layer factors. And a random forest model outputs a teratogenicity score, and both cell viability and teratogenicity score is used and combined to deliver a predictive teratox score. And I will go now into detail of this analysis. But before that, as always, one of the key issues of such gene expression studies is quality control. And we recently published a paper in NAR Genomics and Bioinformatics showing that in public domain, at least about 10% of the data are contaminated simply by wrong tissue. And this can be contamination, this can be wrong labeling. And that's why we perform consequently in our internal studies, gene expression-based quality control with a software that we wrote called Bioactivity. In this case, it revealed that our cells have a stable expression profile and mimics those of very much of the astrocytes as well as neuronal cell types. And in this study, we focused on the germ layer development genes. So there are something around 90 genes of this. And we performed a so-called factor analysis. So what is factor analysis? It is very closely associated with a method that many people know called PCA. And if we take one step back, it is essentially based on expression correlation of the genes. On the left side, I show you the germ layer genes. So these genes has been found in previous studies to be regulated in germ layer development. And you can see that in our system, they show correlation. And these boxes in the middle across the diagonal shows 10 factors, each of them containing one and more genes. And these factors are actually co-expressed genes that are co-expressed upon compound treatment. On the right side, we show you these germ layer factors. As you can see, from left to the right, we have the layer 1 to 3, 4, 5 to 10. And on the right side, I show you a number of genes. And interestingly enough, if you look at these genes and look at their biological information, we realize that these factors are enriched of certain germ layer markers. Factor 1 turns out to be enriched of ectoderm genes, factor 2 mesoderm, factor 3 self-renewal, factor 4 endoderm, and so far so on. And this fact prompted us to build a series of machine learning model using different output, using different features, using different methodology, and using different training test splitting method. And one thing here I want to mention is that a specialty of this machine learning model is that we consider dose response. Therefore, we define not a binary output, but we define a teratogenicity score, which is defined as a cosine similarity of differential gene expression profile with regard to the highest non-cytotoxic dose. So in this case, we transform a binary problem into a continuous prediction problem, and we can make dose response judgment based on dose response. To cut a long story short, we could then for each compound produce such a plot, which we show the viability as well as teratogenicity potential changing by dose. And when compared with the MEST, which is a current standard assay, we observe that Teratox performs more favorably with regard to accuracy and recall. And whereas the precision specificity is still higher in the MEST case. The F1 score, as Jennifer has shown before, the harmonic mean of sensitivity and specificity is higher for Teratox. And more importantly, if we combine the two, we can achieve the best result. Also, we did not only perform the machine learning model, but also we look at which pathways and which biological processes are either impacted by or potentially causing teratogenicity. And there we identified a series of transcription factors that are potentially associated with a change. And last but not least, we performed Bayesian network analysis to build a directed acyclic graph, a DAG model integrating biological knowledge and learning from the data. And we performed sensitivity analysis. And there, what we observed was extremely interesting in the sense that it shows both advantage of our model, namely our model is very sensitive to ectoderm change and self-renewal change, and the limitation of our model, namely it is less sensitive towards endoderm and mesoderm change. This is actually where we want to develop further, improving the model to better characterize different aspects of germ layer development. The summary, Teratox features higher sensitivity and better performance than the current standard mouse cell assay, potentially refining, replacing, and reducing animal research. The success was that the Teratox predicts teratogenicity and distinguishes from cytotoxicity. The challenge is that while it's sensitive for teratogens affecting ectoderm, future work is needed to better model mesoderm and endoderm. And our long-term goal is to replace any models required by regulatory approval. So the Teratox is one story and one project that we are working on. There are other projects of the team that I want to give you a brief glimpse into it. We are developing and optimizing models for toxicity endpoints or assay outcome. We are investing our time in federated learning for chemoinformatics, machine learning models. We are doing toxicogenomics analysis. We perform quantitative DMPK prediction using mono and multi-organ chip systems. We are trying to expand the teratogenicity assessment toolbox. And we are implementing and piloting spatial transcriptomics and machine learning models towards next generation toxicology workflows. And we are performing causal inference of experimental and observational data, as well as setting up computational models of safety profiles of nucleotide acid therapeutics. To summarize, we are a young team working on computational toxicology and safety assessment for preclinical to clinical transition. We perform interdisciplinary research to discover new data types, identify relevant features, and develop tailored models. We collaborate in that process with both wet lab experts and data analytics groups to develop new assays and methods for drug safety. We work towards the goal of causal modeling of drug-induced toxicity for preclinical to clinical translation. With that, I say hi again to the community, and we are doing now what patients need next.
Great, thank you for your contribution, David. Much appreciated. Any comments or questions from anyone? Yep, Nadav, please.
Hi. I have two questions actually, so maybe answer just one, which you see is better, or both if you like. First of all, because you said that the prediction problem is not binary, but you predict the dose response curve, I was wondering how you did that in terms of what metrics you used and what were the challenges. And the second question is you talked about causality. I was wondering how you determine causality as opposed to just correlation. And thank you very much for the impressive work.
Yeah, thank you. For the first question, what we did is following. When we set up the machine learning model, we do a leave one out for the left. So for the rest of the compounds, we first identify the non-cytotoxic concentration. We set the differential gene expression at that time point, at that dose, as 1 if it is a teratogen, or 0 if it's non-teratogen. And for the teratogens, for lower concentrations, we will calculate the cosine similarity to the highest concentration. And with that, you translate the problem from a binary to a continuous one. If there is new question, you can look at our publication, Toxicological Sciences 2022. There you can find the technical details. The second question is a perfect one. So before we heard from Annika about the broad Hill criteria. In this case, in this context, we are meaning from the counterfactual framework as well as the PRO's DAG approach. Now 29th. Tomorrow there will be a now on the AXIF, there'll be a preprint from a colleague of mine in University of Bergen and me writing a review called Causal Inference in Drug Discovery and Development. And there we give a detailed discussion of what we mean with causality in this context. So you're welcome to check out that review. And it's also under review by other reviewers. Once that's published, I'm glad to also share, probably with help of Andreas, the link.
Thank you very much.
Yeah, please send it over because I'm also circulating the cheminformatics newsletter and so on. I'm always looking for interesting papers to include and so on. Also, generally, anyone, just send me those types of information. That's good. Excellent. Thanks, Nadav and David. Any other comments or questions for David? Okay, let's see if there's anything on YouTube. Curious, I don't know which talk was it, this one. Curious if the models based on the binary target are better than with dose response, but I think that might have been from the previous one, right? Yeah, not quite sure.
Yeah, in this case, we tried also binary output, and it turns out the dose response was more useful. If this is happened to be about this talk, yeah.
Okay, yeah. I mean, there's different data sets and approaches, right? And then it's good to learn from different. Okay, yeah, thanks for sharing that. Okay, great. No, thanks a lot, David, again. Yeah.
Thank you, Andreas, for organizing.
Sure, pleasure. Excellent. And now we will move on to AstraZeneca and safety assessment of PROTACs, right? Using cell painting, cell imaging data. And Kevin will talk about this.
Hi, Andreas. Thank you. Can you hear me?
Yep.
Great. Can you see my slides?
Yeah, it's full screen. Looks good.
Looks good. Perfect. Thank you very much. And thank you, Andreas, for the invitation. I'm going to talk about PROTAC safety. But before that, let me introduce myself. So I work for AstraZeneca. I'm a director on safety innovation and PROTAC science lead for the company. So what I will talk about today is it's how do we provide these new modalities for safety mechanism and prediction, and how this cell morphology and in silico methods can help. So I think a disclaimer is that I'm not a data scientist. I'm a molecular biologist by training, but I hope you will find some of the data will present of interest to you. So first, I will start with this slide about what is a PROTAC. So the PROTAC is a degrader modality. So it induces a degradation of your target of interest. And it does this by bringing your target protein that you want to degrade in close proximity to an E3 ligase here. By doing that, it leads to the ubiquitination of the target proteins and a subsequent degradation via the proteasomes. So the PROTAC drugs is actually sitting here in this case. So it contains 3 arms. So the first one has a small molecule binder of the target proteins that you want to degrade. Then on the other side, you have a small molecule binder of the E3 ligase, and these two are linked via linker regions. So the PROTAC is a bit blooming, and it comes from an academic discovery about 20 years ago, but is now in a clinics. So this is from a review that was published recently last year, and showing the compounds that are in clinics or in clinical trials. So the most advanced compounds are from the Arvinas Biotech company, who are in phase 2 with a degrader targeting the androgen receptor for prostate cancer and estrogen receptor degraders to treat breast cancer. So they were in front in leading this PROTAC modality, but now, as you can see in this table, there is about a dozen compounds in clinics or entering clinical trials targeting various proteins. So what about PROTAC safety? Why so far what's been reported from the companies that are in clinics, the compounds are well tolerated, but it doesn't mean that there is no safety liabilities, and I will introduce some of this in my talk. So the things we know is that there could be safety liabilities associated with some of the chemistry used to bind the E3 ligase, which is represented here. So again, as a PROTAC mechanism, 3-part, a binder to the E3 ligase, which is here, then you have a binder to your target protein, then you have the linker region. So if you look at the previous tables, and most of the compounds that are in clinics uses the cerebellum E3 ligase to induce this targeted degradation. And they do that because they hijack this, or they use these small molecules that binds to cerebellum via molecular glue mechanisms. And these drugs are very well known. The field which has just been introduced to thalidomide here. So the PROTAC community has used these compounds to bring the target protein close to cerebellum for degradation. And this is, for example, the structure of the AR degraders, called ARV110, and it's using this pomalidomide derivative here. And this is the structure of the ER degraders, which is using a lenalidomide warhead here. So the use of these drugs could come with some safety liabilities, and that's for a few reasons. First, these drugs are approved drugs, so we know their safety profile very well. So it is possible that the toxicity seen with these drugs could translate into the full PROTAC molecules. So what do we know about these compounds is that they're actually behaving like a mini PROTAC machinery by themselves. They induce the degradation of proteins, but it's not via what is called a PROTAC mechanism, but what is called a glue mechanisms. But this is some of the transcription factors that has been shown to be degraded by these IMiDs drugs families, like thalidomide, lenalidomide, and pomalidomide. And it's only recently that we can start linking the toxicity seen in clinics with the degradation of these transcription factors. And I will touch on this in the next 2 slides. So in clinics, what we have seen with these drugs, that the dose-limiting toxicity is bone marrow tox or neutropenia. But there are also toxicity seen with these drugs, like peripheral neuropathy or some cardiovascular thromboembolic complication here. So it is possible, in conclusion, that the PROTACs based on IMiDs may carry exactly the same liabilities. And so we decided to start monitoring the degradation of some of these transcription factors. So we just discussed about the teratogenicity, and the link between thalidomide and teratogenicity has been more clear over the last few years, and that's by the degradation of SALL4, which is some transcription factors involved in the differentiation of these stem cells. So SALL4 is these zinc finger proteins and induce the differentiation of pluripotent stem cell into somatic cells here. And SALL4 is recruited by IMiDs to cerebellum, and that induces this degradation. So that's a cartoon represented what's happening here with the IMiDs drugs. The SALL4 is going to the cerebellum complex for degradation, and then degradation with the proteasome, and that's causing teratogenicity and myopathy. Doesn't mean SALL4 is the only transcription factor involved in this event, but it's one of the main proteins that has been validated. So we are monitoring the sulfur degradation and by Western blot in this case, and we were able to confirm that the sulfur degradation potency with this with the image drugs here, and the thalidomide is the least potent of all, and pomalidomide is the most potent of this sulfur degrader. And we saw translation when there were image-based PROTACs or pomalidomide or thalidomide-based PROTACs, which is in this Western blot here. The thalidomide is here a dose response from 0.1, 1, and 10, but this is a PROTAC based on image, and it shows also degradation of sulfur at 1 and 10 micromolar. So there was translation of the sulfur degradation to the full PROTAC molecules. So we are using sulfur as a potential good biomarker for the developmental toxicity, but we need to understand a bit more about the translation in vivo. So regarding the bone marrow toxicity or the neutropenia that I mentioned, which is a dose-limiting toxicity for these drugs, this has been associated with the degradation of the Icaros transcription factor or KZF1. So KZF1 is also recruited to the cerebellum by IMiDs and uses this degradation. So Icaros is actually expressed in the hematopoietic stem cells and is increased expression across the differentiation. And the literature suggests that with the IMiD drugs, there is a block of the differentiation at the myeloblast level because of the Icaros degradation that in turn regulate the PU1 transcription factor. We also confirmed by Western blot that our PROTACs based on IMiDs can degrade Icaros in this Western, and sometimes as good as lenalidomide, or sometimes even better than lenalidomide. So this is another biomarker that we are following, but this time for bone marrow tox prediction. Now, this is what we know about it, and we focus some effort with bespoke degradation assay to look at this. But like for every drug, there are the things that you know and the things that you don't know. So how do we tackle the things that we don't know? And thanks to this meeting, now we've highlighted the role of big data and the high throughput or high content systems to try to screen compounds at scale and predict the drug response. And that's usually done with multi-omic technologies, but wouldn't be able to analyze big data without data science, machine learning, and AI. So these three comes together. So I'm going to introduce one of the assays that we have tested in AstraZeneca, which is cell painting, and we have been introduced to cell painting as well today. But just as a recap, the cell painting, it's an imaging assay. It has many different dyes to label different compartments, and then the machine learning will look at the morphological changing in all of these different channels, and then to generate the signatures of the compounds. And then you can play with the signatures to make some chemical clusterings activity and to predict the drug response if you have a library. But the bottom line is that you generate the signatures, compound signatures, and you interrogate the signature between each other. So we have tested the cell painting with PROTACs and using more than 500 compounds. And we have been able to do three things mainly. So the first one is to measure a cell painting activity score or that we call a grid score. And this grid score basically look at the difference of the compound's effect compared to DMSO, but it integrates all these different features that you can extract from the cell painting data. We also tested non-PROTACs as well. Either it was some as an individual component of the PROTAC molecules, or it was approved drugs as a control set. So with the PROTAC, we were able to see a dose response activity in the cell painting activity score, and from 0.1 to 1 and 10, where there were 80% of the compounds were active at 10 micromolar. For the non-PROTAC, we were a bit in similar ballpark. There was a dose response increase in the activity. So that by itself doesn't tell you what the drug has been doing. It's just telling you that there was an activity detected in the assay. So an interesting thing that you can do is the chemical clusterings, and in this case using the UMAP plots. And all the compound tested were from about 15 projects, internal projects. And we are very pleased to see that we could see differentiation of the activity across projects. And we are highly biased towards one particular project, and this is because we had some we had some activities that we wanted to monitor, and I will talk a bit about this. But for example, there was a cluster here with this, the BRD4. These were compounds that were published in the literature here. So we knew we had something we wanted to check with this cluster here. We knew in these programs we have some activity in on mitochondria. So we had some mitotoxicity signals, and we wanted to see if that was detected by a cell painting assay. So we did annotate this UMAT plot using a mitotoxicity assay, which use the FG2 cells growth in galactose. And that's where the cells rely on mitochondria for energy productions. So these cells in this condition are highly sensitive to mitotoxic insult. And we're very pleased to see when we annotate these compounds that it was indeed this cluster that shows the highest mitotoxicity score, which is in purple here. Now, what was also interesting is that there were compounds that had no annotation, which are in gray. So we had no data in the FG2 galactose assay, and we built a model, a machine learning model, to predict that these compounds will indeed be mitotoxic. So we have done the other way around. We took these compounds from the cell painting data and put them in the galactose assay, and the prediction score for mitotoxicity was very good and very high. And this has been published now in the SES Chemical Biology. If you're interested, the link is here. So we wanted to understand a bit of the nitty-gritty of the dataset and see whether we could drive the SAR with the cell painting assay. And that's an interesting observation with the PROTACs here. So we did screen the binder of the E3 ligase, and we also screened the binder of the target protein of interest. And they were inactive, mostly in the cell painting assay. But when you put these two together and form the PROTAC molecules, this is the PROTAC molecules that were active. So it's an interesting observation in this new modality that the prediction or the activity couldn't be predicted by the individual component. So it raises the question of of whether it is worth profiling the individual component to predict the full PROTAC drug response. On the other plot here, we did characterize some published compounds, PROTACs, and small molecules, and we are also able to see like the cell painting could detect various activities with different compounds. And for example, as particular, this compound here is the CDK9 degrader. And when we look at the raw images, we're able to see that in the treated samples, there were a loss of nucleoli, which is in line with the block and cell cycle progression. So that's in line with the on-target biology. That's to show that there is many different activities that could be detected by a cell painting beyond the mitotoxicity. So with that, I would like to acknowledge all my colleagues that have helped building this data set, and I would like to thank you for your time, and happy to take any question.
Yeah, thank you, Kevin. Right, there's always the question of the small molecules over decades, over 30 years, whatever, figured more or less out how to profile them. Yeah, doing safety profiling panels and how to anticipate FUSCAM, understand that, right? Now you have new modalities, so PROTACs, and now you need to start all over again and understand, well, both FUSCAM as well as more bioactivity related aspects, right? Yeah, but good to see that cell painting in this area can help as well. Okay, any questions or comments from the audience? Yep, another, please. Yeah.
Well, me once again. I was wondering if you could maybe share a metric for like a quantitative measure of how good your prediction was, especially maybe when compared to an alternative. I mean, how can you see that this cell painting has given you an advantage over other things? Thank you.
Yep. Thank you. So I think this is part of the publication, so there will be more detail about the exact numbers. The prediction was very good. I think it was 80 or 90% prediction, if not more. But it was a small data set, so I think we need to build the data set to increase the numbers here.
Thank you very much.
Yeah, cheers. And Caroline, do you have a mic, or should I read the question here?
Hi, Kevin. I was just wondering if because you mentioned omics data sets in one of the slides, and I'm just wondering if you investigated the omics technologies, just like proteomics, and if for the compounds you had also in the cell painting, and if these data sets showed different, similar groupings for the different technologies for the compounds.
Yes, Carol. That's something we want to do. We haven't done it yet, but that's planned.
Okay, thanks.
Thank you. Any other comments or questions? No, just checked on YouTube. There's nothing. Okay. Excellent. Great. Thanks a lot. But very good to see that there are investigations to find out about the applicability domain of those new approaches. Yeah, because only then, if we know where we can use them, we really understand whether they're applicable in the future. Right. Okay. Great. Excellent. Oh, there's one more. David, please.
Thank you. It came too late to me. When I look at the publication, it seems that you're down to 100 nanomolar. I wonder if you go still down, can you see still these subtle changes, or is it you need quite high concentration to see such morphological changes?
Good question. We haven't tested lower than that, but it's a good question if you want to understand your real potency. So I would say at 100 nanomolar, we're at 16% of the drugs that were active in cell painting. I think if we go down another 10 times, there will be a reduction, but that may be where we capture maybe the primary pharmacology versus the secondary pharmacology. That's something we need to understand better and deconvolute. It's a good question.
Thank you.
Okay, great. Thanks a lot. I have a student who's also treated who's analyzing cell painting data, because the question is always, you have quite a high dimensional feature space again, right? You don't necessarily want to select features because then interpretability might go down. Yeah? And there are ways to put them into like an SOM, self-organizing method similar to that, to actually organize them, to kind of cluster them in order to have regions that tell you about the mode of toxicity of compounds. I think next time also we can present that as well. Okay, I think there's no further comments or questions. Great, thanks a lot, Kevin. Yeah?
Thank you, Andreas.
Cheers, everyone. So yeah, now we come to a company that is not quite the size of AstraZeneca just yet. There is AbsoluteAI, where Layla Hosseini-Gerami will present about approaches they are taking to predict drug safety. Can you share your slides as well?
Mm-hmm. Sure. Can you see that okay?
Yeah, looks good and sounds good.
Okay, great. Thanks, Andreas. So I'm Layla. I'm a PhD student in Andreas's group at the University of Cambridge, and now moving into my new role as co-founder and machine learning lead at AbsoluteAI. And today I'm going to talk about in silico toxicity prediction and its application in preclinical drug discovery, and specifically talking about where we are now and how we can make progress in the field. So today I'm going to be covering toxicity screening and where it currently fits into the drug discovery pipeline. I'm then going to introduce the concept of proactive high volume toxicity screening implemented using in silico tools, which is what we're trying to do here in the company. So we're a very early startup, as Andreas mentioned. So we've had a lot of conversations with different people in industry, from big pharma to small biotechs and CROs. We've been able to identify what their pain points are in applying algorithms for in silico tox prediction at an early stage. And that's kind of going to be what the talk is mainly about, these problems, and then also the solutions that we've identified to address these problems, which we are building into the algorithms that we're developing currently. And then leading on to a discussion on future prospects to advance the field, and kind of opening up the discussion to this in silico tox community, as well as communities of non-in silico scientists, to really try and understand together why aren't these tools being applied more widely in industry and how we can address these issues. So currently, safety screening happens mostly towards the end of the drug discovery process. This is because experimental screening is time-consuming and expensive. Particularly, this is evident for smaller biotechs who are limited in resources and need to outsource these experiments. So they really can only perform safety screening at the end of the pipeline when only a handful of compounds remain. As we all know, this has knock-on effects, leads to high failure rates in the clinic, and very expensive and unsustainable drug discovery. There are in silico tools which are being applied in industry to predict toxicity early, which we've been seeing today. But what we'd like to do at the company, and where we feel the field is moving towards, is this concept of resequencing drug discovery and providing proactive screening right at the start of the drug discovery process using in silico tools, so that safety problems can be detected and mitigated against right from the start of the drug discovery process, leading to fewer failures, cheaper screening, and more sustainable drug discovery. So this is possible now because in silico tools are starting to match or surpass high throughput in vitro methods. This is seen in the literature, but also with our internal mitotox prediction tool. So we've here compared it with 2 high throughput mitochondrial toxicity in vitro assays, showing in terms of balance accuracy and Matthews correlation coefficient that we're in fact surpassing these in vitro assays. So we're currently partnering with a few different companies to give them access to our technology and also to be able to validate our predictions in real life situations. So a case study of how this is being used, this proactive screening is being used, is with a biotech who we're partnering with. They have a high throughput screening output of about 55 hits. Using kind of chemical filtering, traditional chemical filtering, and kind of chemist intuition, they've managed to narrow this down to about 20 hits, but from this point on, it's very difficult to distinguish them, and they can only take about 4 or 5 series forward. So now using our technology, they're able to filter these 20 compounds to produce a list of compounds at the end, which will hopefully not lead to any safety problems later on in the clinic. So from our experience, these small companies such as biotechs are more likely and more willing to adopt this kind of approach. On the other hand, talking to big pharma, they're understandably a lot more resistant to this kind of resequencing approach and this high volume proactive screening at the start of the drug discovery process. And from what we've been hearing from these people, it's not just about accuracy, although model accuracy is important. What's really important is to build models which medicinal chemists can trust, and that provides some value in the decision-making process, because if these methods are implemented early, we need to be able to give them guides to make decisions. So i.e. not just using black box yes or no models. So I'm going to be talking through some of the problems that we've identified and the potential solutions as well in the next few slides, before I then kind of invite the audience to give their views as well. So the first problem is that the chemist doesn't know whether their model's prediction is reliable for their compound series. So we can report very high accuracies on external test sets, but this doesn't tell us the full story. External test sets can be constructed in a way that is very biased, and indeed the data going into these models can be biased themselves in terms of areas of chemical space, for example. So one possible solution to increase this trust and to give some kind of idea to the chemist of whether or not the prediction is reliable for their particular compound series is applicability domain analysis. So the applicability domain defines the areas of chemical or biological space, depending on which features are used to build the model, whether predictions are reliable. So an example of this is called reliability density neighborhoods, which creates a map of chemical space considering the local density, which is the number of nearest neighbors, and the local reliability, which is the precision and bias of training instances. And here you can see it represented as a schematic, where we have areas of chemical space which are denser and more reliable, and areas which are sparser and less reliable. So unseen compounds can be mapped onto this RDN map, and then that can be used to basically assess how reliable the prediction is for this new compound. So to illustrate how this works in practice, I've taken a compound here, which is a melanin concentrating hormone 1 antagonist from PubChem. So this RDN analysis is incorporated in the Pigeon V4 target prediction tool, which was developed in the vendor group by Dr. Lewis Mervyn and then retrained by myself and Mariana Trapozzi. So this tool is essentially a collection of random forest models built on bioactivity data to predict the probability of activity of different targets, but also incorporating this RDN score of applicability domain. And it's also possible to perform a similarity analysis to retrieve the nearest neighbor in the training set used to build the model, as well as the Tanimoto similarity, which is here in terms of ECFP4 fingerprints, which is what was used to build the models. So looking at this example here for this compound, we can see the top predicted target is the known target itself with a very high applicability domain score. And we can see why that is. It's because the nearest neighbor is the input compound itself, Tanimoto similarity of 1. The second highest predicted target, which is here an epigenetic modulating enzyme, is again predicted with a very high probability, but the applicability domain score in this case is a lot lower. Now, when we look at the nearest neighbor in the training set, we can see that there is some similarity, but it is quite a different molecule. So this can be used, this information can be used so the chemist can decide whether or not they really trust this prediction or not. The third highest predicted target is one that we all know, which is hERG, again predicted as active, with a relatively high applicability domain score. And when we look at the nearest neighbor, we can see that it's actually extremely chemically similar to the input compound, in fact, part of the same chemical series with a higher Tanimoto similarity. So you can see how providing these three columns, as well as these two columns, really provides this extra information to help the chemist make their decision on whether or not they trust the prediction or not. So the second way to address this issue is conformal prediction, and we've already had a nice summary of conformal prediction from Jennifer. So this is a framework which sits in front of a traditional machine learning algorithm to provide mathematically valid confidence estimates for predictions. So how it works is a prediction interval is calculated for each predicted object at a specified confidence level, and this is based on the non-conformity of the predicted object to the objects in the training set. So this is shown here, an example from the paper, predicting the log D for omeprazole. And you can see here how we can put the reliability in the hands of the chemist. They can increase the confidence score depending on the stage of the drug discovery process. And they get as an output, instead of a single prediction, an interval of predictions. And then that can be used to, again, decide how reliable is the prediction for this specific compound, and can we trust it to, for example, throw it away at a very early stage in the drug discovery process. The second problem is that a compound might be toxic and a model might pick it up as toxic, but only at a dose which is far higher than the efficacious dose. And of course, the chemist doesn't want to be throwing out these compounds right at the start. So one way to mitigate against this is quite a simple method, but it's just to build models at different concentration thresholds. So here I'm talking about in vitro concentration, but building these models in a more granular way can be used to extrapolate using PKPD into in vivo dose. So having a model which is just one binary classification model and arbitrary threshold can't be used to then make these decisions and to extrapolate and understand the relevance in vivo. So here we've got some in vitro PIC50 data for a toxicity target, which we've represented in 3 different types of models. The first is regression, which is just directly predicting the PIC50, which is very preferable. But in practice, this can be quite difficult to achieve high accuracy for because of the noise and bioactivity data sets. So it's often preferred to perform a classification. So one way is performing a multi-label classification, where we can classify compounds as either non-blockers or weak, moderate, and strong blockers, depending on concentration thresholds, or simply building multiple binary classification models at different thresholds. So you can see here, we move from 1 to 10 to 30 micromolar, where we define a compound as toxic or non-toxic. The ratio of non-toxic to toxic changes drastically. So building on a bit more about how we take these in vitro predictions to in vivo. So obviously a lot of in silico tox models are built on in vitro data because it's far more abundant, there are fewer confounding variables, etc. So we really want to start moving from predicting these in vitro toxic endpoints, which are, after all, just a proxy for in vivo toxicity, and really start to build up a whole picture of a whole organ holistic tox model. So we're already starting to consider an in vitro in vivo extrapolation. We've seen a lot of talks today considering models' predictions in terms of in vivo relevance. There's also advances in adverse outcome pathways, which are being integrated with aggregate exposure pathways, which essentially are then able to identify the likelihood of a molecular initiating event happening based on predicted exposure. But as well as this, other types of data can be integrated to build up a picture of in vivo organ tox. And these data are becoming more and more abundant and available nowadays. So for example, species, tissue, and target expression is the off-target of interest expressed in the tissue or the organ. And is this expressed in different species, for example, human, rat, dog, or whatever the animal model may be. Using histopathological and clinical observations to relate these in vitro findings to in vivo clinical findings, and also omics data and biomarkers, especially now as we're moving towards the era of personalized medicine, using genomics data to identify SNPs, which might make a patient more or less susceptible to compound toxicity, and again, having this information about whether a compound will be toxic in certain patient populations. So the third problem is that it's very difficult for chemists to trust a prediction that's coming from a black box model. With an experiment, something that they can see and they can touch, but a machine learning model, they really want to know, why is my compound toxic, before they can trust what's coming out of it. So models which are used in the decision-making process have to be interpretable. So these complex deep learning architectures are very popular. They show very good performance, but they are usually a little bit harder to interpret. We've seen some good work from Moritz on how to interpret these methods, but in general, it's much easier to use something like a tree-based model, which are inherently interpretable, but these are often thought to be less accurate than the deep learning models. So my suggestion here from experiences with the company and also during the PhD is don't crack a nut with a sledgehammer. Run some baseline random forest models alongside some more complex deep learning models if possible, and assess whether there is a performance gain when using more complex models, and whether the performance gain is an added value over having a model that is inherently interpretable and that a chemist can actually use and follow to understand why their compound has been predicted as toxic. So in terms of what we're implementing here in the company, we're using graph convolutional networks, which is a deep learning method, but can be used to interpret contributions of different atoms to the toxic prediction. So here we have an output where we've applied a model to this compound here, where we have, again, similar to what Moritz was showing, areas that are contributing to the toxic prediction and areas which are not contributing to the toxic prediction, and how this adds value. Again, coming back to value and decision making. A chemist can see whether a part of the compound which is contributing to toxicity is part of the core of the compound that is required for efficacy, or whether this could be an R group which could be switched out. And the beauty about computational models is that we can perform R group enumeration and feed these compounds back into the model very quickly and be able to tell the chemist whether or not they can modify their compound to mitigate against any safety issues. Furthermore, we found we're using feature importance plots. It's a lot easier, it's more informative to use more interpretable features such as phys-chem descriptors, although those aren't always perfectly interpretable, compared to using something like hash molecular fingerprints, which can be difficult to then go back and map to the original chemical structure, some chemical substructure. So another way to mitigate against this issue is to, as well as using machine learning methods, complement them with inherently interpretable non-machine learning methods. So this is using things like omics data, which again is becoming more abundant and can be used to investigate mechanisms of toxicity, which we saw, for example, from Annika's talk. So there's a class of algorithms called causal reasoning, and they use transcriptomics data and sign-directed prior knowledge networks to reason upstream and find signaling proteins and pathways which are responsible for the transcriptional effects. So one example of causal reasoning algorithm is called Carnivore, developed in the SAYS lab in Heidelberg, and together we have implemented it into a GUI app called Maven, which is available on rShiny. We have used this to apply this to transcriptomics data derived from hepatocytes treated with a high dose of acetaminophen, which, as we all know, causes drug-induced liver injury, and a hepatocyte-specific prior knowledge network. And what Carnivore has done is it's optimized a dysregulated signaling network capturing the perturbation of acetaminophen on the cells. So the resulting network, visualized here in Cytoscape, is highly enriched for the interleukin-17 pathway, where the nodes are highlighted here in green, and this pathway has a pivotal role and is known to play a pivotal role in acetaminophen-induced hepatotoxicity. So providing this information alongside predictions is very valuable, again, so looking at the mechanisms of toxicity, but also potentially ways of mitigating toxicity. For example, interleukin-17 signaling has been suggested as blocking it has been suggested as a way to mitigate against acetaminophen-induced toxicity. So then how do we improve the field and how do we kind of progress forward together? So some solutions that we've come up with is building trust and confidence with experimental scientists. So talking to them and listening to what they want from models and what their limitations are, and also us talking to them about what the capabilities of our models and what we can do and sharing information together. The second aspect is companies being more willing to share their data. For example, pharma companies, where they have data where their drugs might have failed and then because of toxicity, sharing this so that we can develop models which cover better chemical space and will in the end benefit all of us. And the third aspect is further developments in these kind of in vitro and vivo extrapolation techniques and building models which are able to predict in vivo responses. So before I say thank you and invite discussions from the audience, I would like to give out a call to collaborate. So we're looking to build on existing partnerships and consortia across drug discovery to tackle the big issues that we face. Together we can create better predictions earlier, ultimately saving lives. So if you're interested in collaborating or learning more about what we're doing, then feel free to send me an email. I want to thank you all for listening. I also wanted to acknowledge BBS and Eli Lilly and Company for funding the PhD work that I carried out, of which some was in this presentation. Thank you very much.
Yeah, very good. No, thanks for presenting, Layla, and also good to see that startup companies move in that area, right? I mean, there's a lack of data, I agree with that, yeah. But also, how do we use machine learning, predictive tools, yeah? Well, the decade that started, whatever, over the last, the direction that started over the last decade or so, yeah, AI, ML, I mean, that's not always productively used in the field, right? Because there's data shortage, right? Interpretability, you have legislation, right? What do you need for, from that aspect, actually? It's actually difficult to find an overlap and find an approach that satisfies all the needs that we have. No, but very good. We wish you both for your company, but also for the whole field and best of success. Yes. Any questions, any comments from anyone? Okay, let's see if we have something on. Okay, no, good. Can you actually share some of your current thinking in the sense of what do you want to do differently? Because many of the questions you post, they are, of course, generally true. Interpretability and at what stage do we use predictive models? What do you want to do differently? Can you outline that? Because that's always the question, right? But what is new if you have a startup, right? I mean, then what do you do differently? How would you describe that?
So I think the main novelty of what we're trying to do is this early prediction. And again, like I said, the way that we're hoping to allow this to happen early is by implementing these things that I talked about today. So incorporating not only the black box yes/no predictions, but having the interpretability, applicability domain, and really building tools that add this value for the guidance so that they can deploy them very early on and have the confidence that they'll be able to follow these through without failure later on. So what we're trying to do is kind of gather all these different aspects of models that we're seeing better interpretation methods, better applicability domain methods, conformal prediction, and essentially trying to build tools that are practically useful, which we believe currently is lacking.
Yeah. One important question is, of course, you have a prediction. What then, right? So how do you follow up by assays, for example? I mean, you have the model, right? But then you also have the decision-making process in a given context, a given project, a different type of chemical space, a certain disease, and so on, right? And those interfaces, in my experience, that's really what I learned at Astra. That's very important as well, yeah? I mean, the model says something, okay, what comes next, right? Imagine you cannot do an essay, so what do you do, right? So basically the interfaces at the front, at the back, so to speak, yeah? What's actionable information, right? And that's something that's very important to understand as well. It differs hugely depending on the context, yeah? Do you have an essay, which disease area is it actually, yeah? I mean, that really makes a difference. Okay, great. Doesn't seem there are any questions if I see that correctly. Great, thanks a lot again, Layla.
Thank you very much.
Great, thank you very much for your invite, Andreas. And today I will present about our work. So our approach is to toxicity prediction is quite different in the sense that we took a hypothesis-driven approach, which is quite aligned with Andreas' recent two-part review on AI in drug discovery and development. So first of all, and in the past 10 years, we spent $10 million to build what is called the virtual human platform. It's actually the aggregation of more than 150 precise models of human diseases, as well as a model of human physiological aging. So all of these models are actually white box knowledge graphs, which are highly interpretable, and which enables this platform to be the functional reconstruction of human physiology and human pathogenesis, thus enabling early and accurate prediction of clinical efficacy and toxicity. So our approach was actually motivated by observing the the long-term low clinical success rate, especially for first-in-class novel medicines, about 5 to 10%, 5% to 10% across the board since 1980s. And what's very interesting about this problem is that in the past 40 years, actually the whole world witnessed a boom of biotechnologies and the new types of data, but apparently these new data did not really significantly alleviate the problem low clinical success rate. So that actually touches that the early predictions of clinical efficacy and toxicity may not be performing very well, despite these new waves of more data. So our methodological assumption based on this observation is that it might be not the amount of data that's a bottleneck in clinical toxicity efficacy prediction, but instead it might be there might be some systematic bias and a conventional bioinformatics approach towards understanding or labeling biological data that might make top-ranked novel drug targets usually concentrate on the downstream effectors of disease etiology, especially for heart diseases like age-related diseases. So based on this methodological assumption, and we started 10 years ago to build a pipeline towards reanalyzing biological data, specifically labeling biological data in a highly comprehensive but detailed manner in order to build white-labeled knowledge graphs for clinical efficacy and toxicity prediction. So very importantly, we took a first principle-driven approach by leveraging the perspective from computer science, especially from coding. So if we look at disease, drug, and efficacy and toxicity, all of them have their counterparts from a coding perspective. Like disease could be viewed as a software bug, and a drug could be viewed as a software patch. And efficacy basically answers the question, does the patch fixes the bug? And finally, toxicity answers the question, does the patch cause fatal errors that's unexpected? So from the computer science perspective, especially from coding perspective, so toxicity cannot be fully understood or even predicted out of the context of the efficacy. So that's the reason why that we came to this first principle in our approach, which that toxicity prediction must be made in the context of efficacy prediction. So driven by this principle, and we further leverage from the success in other similar scientific disciplines, for example, physics. Physics and biology, they're quite similar in terms of the complexity of their research subject matter. For physics, it's a multi-scale, complex dynamic system, the whole universe. And in biology, it's a life, which is also multi-scale, nonlinear, and complex dynamic system. And physics is a very mature discipline since in the past century. And one defining characteristics of physics scientific approach is this synergistic combination of top-down theory-driven approach and the bottom-up data-driven approach. And more predominantly in physics, it's usually the case that theoretical physicists, the first and the foremost, came up with comprehensive but detailed, rigorous theory and made lots of predictions. And then experimental physicists start to build experimental infrastructure and collect the data over a long period of time, about a few decades, in order to, and most of the data finally confirmed the predictions made by the theory. However, in biology, this whole top-down theory-driven part is missing. That's actually inspired us that we need to fill in this gap by carefully understanding and labeling biological data. And by labeling data, we actually specifically mean to come up with hypotheses about the functional roles of all the building blocks implicated in human physiology and in pathogenesis. So there are three types of hypotheses or three types of labels that we assign to biological building blocks. The first type is called a functional role hypothesis. It basically refers to the specific function that a specific building block plays in normal physiological context. So by building blocks, we refer to basically as so from the on the genetic side, and what is the functional role of a specific nuclear base, and the functional role of each amino acid type, and proteins, pathways, cells, and tissues, and also organism species specific functions. But that is just the step one. And then on top of which, we add another type of label, which we call algorithmic role hypothesis. Algorithmic role hypothesis specifically refers to the functional role played by each biological building block, directly or indirectly, in the process of aging. Because from the coding perspective, aging is a ubiquitous phenomenon across all life forms on Earth. And so that's the reason why we must be able to leverage, especially cross-species biological data that are publicly available, in order to tease out the functional role played by building blocks in aging, because most of the diseases of all highly unmet need are age-related diseases. So these are the first two types of labels about the normal functional role played by biological building blocks. And last but not least, we also add a third type of label, which is called clinical role hypothesis. So by clinical role hypothesis, we specifically refers to the role played by biological building blocks in specific disease etiology. More importantly, we clearly label that whether, for example, a specific pathway, it is an upstream driver or a downstream effector in the context of a specific disease etiology, like for Alzheimer's disease. So for each biological building block, we assign these three types of labels or three types of hypotheses. So that's basically the pipeline that we have developed to batch generate a large number of novel biological hypotheses that have clear clinical relevance. So after we have generated hundreds and thousands of novel hypotheses, then the next important step is actually to falsify or to validate those hypotheses. So because our platform was motivated to make early, accurate predictions of clinical efficacy and toxicity. So that's the reason why that we cannot just rely on cell lines or animal models for the validation purpose. So that's the reason why that we came up with a novel approach of making predictions of ongoing pivotal clinical trial outcomes as a systematic way to evaluate the clinical scale quality of all these novelly generated hypotheses. If the predictions can be of really high accuracy and precision, then our batch generated novel biological hypothesis would become batch validated clinical scale novel knowledge. So applying that approach over the past 10 years, we have developed our platform in full, now with 150 models covering all major human diseases, as well as one foundation model of human physiological aging. So this platform is literally a virtual human platform, because as we said, as the name suggests, it's a functional reconstruction of human physiology and human pathogenesis, but still in a quite abstract, interpretable way. And this platform enables us to simulate drug-body interactions in a highly precise way, such that we just need to take into account two types of inputs. The type 1 is a drug mechanism of action input, which has 4 parameters, which is the drug target modulation, and drug modality, and the drug delivery mechanism. Another group of input is the clinical trial design protocols, including the inclusion-exclusion criteria and the primary endpoint choices, control group designs, and indications. So with just these two groups of input, we could actually run a virtual pivotal clinical trial in order to evaluate the clinical efficacy and toxicity for such a combination of parameters covering the drug as well as covering the patient's characteristics. And we can then give very clear evaluation regarding whether it will achieve clinical success or not. By clinical success, we mean that at least one primary efficacy primary endpoint would be met, without major toxicity issue. So that actually reflects our philosophy that the preclinical toxicity and clinical efficacy, they must be predicted in a coherent manner. And when it comes to the final important validation that echoes Andrea's reviews perspective, that basically increasing clinical success rate could be like a final verdict for any platform that could help improve toxicity prediction. So that's actually the same spirit behind our prospective validation process. So the one traditional way that given the platform, we could start as many clinical trials as possible, but that would be economically and temporally infeasible. But alternatively, as you may see from AlphaFold 2, another AI-driven approach, we are inspired to just predict the outcome of pivotal clinical trials sponsored by all biotechnology, biotechs, and biopharma all over the world, and with the first-in-class preferred without limits on any indication. So in this way, there are actually 300 new trials every year that actually very importantly can enable us to reach 131 trials as a minimum requirement for 90% confidence interval. But this validation is equally effective compared with starting lots of the trials by ourselves. And more specifically, the entire real-world validation has been the most rigorous so far because it's 100% prospective, only use trials without any readout available as of yet when we make the prediction. And also, very importantly, the entire validation is public. We publish the prediction of a clinical outcome well in advance using Twitter with clear timestamps showing that we made a prediction before the company first released the readout. And also importantly, this validation is an external validation. By external, it means that the data that we use to build the platform has very little overlapping with the data to test the platform. Because our platform was built from understanding and labeling publicly available biological data, we did not use any past historical clinical trials to train our platform. We reserve all the clinical trials just for testing the platform. That's the reason why that our validation is external validation that could reflect the true predictive power when the platform is confronted with new indication or new patients without any historical clinical trial data. And last but not least, so this validation has been the world largest scale so far. Because in the past 2 years, and we have published 265 pivotal clinical trial predictions covering phase 2 and phase 3, and without limits on any medications. So this number, and also for the past 2 years, we have waited to have 173 readout events to become publicly available. So both of which has met the minimum requirement for 98% confidence interval. And very importantly, when we make a prediction using a platform, we only use publicly available data like the clinical trial design protocol from clinicaltrial.gov and also publicly available MOA data for the compound. So our performance has been really outstanding because you can see that we achieved 84.4% prediction accuracy, covering both cases of clinical success and clinical failure, which has reached 99.9% statistical power. And in contrast, the runner-up company of the world with a similar platform has only published 6 pivotal clinical trial predictions so far with 50% accuracy, no statistical power. And more importantly, we look at other performance metrics, especially look at precision. So this is only concerned with the percentage of correct predictions of clinical success. We actually achieved 79.0% for first-in-class and drug indication pairs. And this has a clear industry benchmark because the rationale is that every medicine that enter the human clinical stage is actually reflect a clinical success prediction made by a drug developer after careful review of all the preclinical data. And then the percentage of all clinical stage assets finally finish all clinical trials and got a marketing approval is actually a measure of precision as a percentage of correct prediction clinical success made by drug developers in the conventional best practice. So historically, in the past 10 years, this has been about 6.3% for first-in-class drugs across all indications. So this actually shows that with fewer amount of data, actually our platform can improve the precision or the what we call the clinical success rate by 12.5-fold. This is also done across the board without limits on any indications. And more specifically, if you look at our validation, and out of 173 trials with readout, 131 of them are first-in-class, and 133 of them are first-in-class, and 131 of them are age-related. So both of these subgroups representing the most valuable, but also most challenging groups for clinical predictions. So we have achieved both more than 10-fold improvement in clinical success rate. And we actually use, when we make these predictions, we're actually at a data disadvantage position because we only can have access to the publicly available relevant data in contrast to each drug developer has their proprietary data to aid their important go-no-go decisions. But as you can see that our platform has been well validated in an unbiased way across all therapeutic areas. As distribution of trials in our validation per therapeutic area is not different from the distribution of trials across started worldwide since 2020 in different therapeutic area. And our performance has been very consistent across different therapeutic area, especially for the hard area like oncology and neurology. We have achieved, actually we did even better, especially for oncology. We achieved nearly 16-fold improvement in clinical success rate compared with the appropriate benchmark. We also listed a more conservative benchmark, which is a historical likelihood of approval from phase 2. And the reason why we list this is that in our validation, we only use pivotal clinical trials, but actually the data we used to make those predictions were available at the preclinical stage. So that's the reason why we listed 2 benchmarks, and we outperformed both of these benchmarks by a large margin. And very importantly, our platform has been able to evaluate the clinical efficacy and toxicity of different drug modalities. So far, we don't have any limits. We can evaluate molecules, biologics, and even PROTACs and RNA therapy and the different kind of vaccines. So we also did the correlation analysis to try to find out what are the major contributors to this across the board drastic improvement in prospective clinical success rate, especially in the accuracy of prospective clinical efficacy and toxicity prediction. So as you can see that the correlation between the F1 score of our predictions and the historical clinical success rate is actually a weak and negative correlation. So this actually means that our improvement, if we can view the historical clinical success rate as a measure of the existing knowledge in literature that can be leveraged for drug discovery and development, our improvement is not related or correlated with existing knowledge. So that actually confirms our original approach that by reanalyzing existing biological data, because we don't have any access to any exclusive data, we actually discovered a large amount of novel clinical scale knowledge from existing biological data that enables a drastic improvement in the accuracy and precision of clinical efficacy and toxicity prediction. And also the correlation is a little bit negative, which means that for the harder therapeutic area like oncology, age-related diseases, and the quality and quantity of knowledge we extracted from existing biological data, it turned out to be better in clinical predictions. And very importantly, when we say that it's important, given that you have achieved pretty accurate predictions, but how early can you go? Do you need or depend on available, like when you make phase 3 trial predictions, do you need to access to the phase 2 data? That's not the case for our platform, because we are actually doing drug-body interaction modeling in a de novo manner, which means that we do not rely on whether there are prior clinical data or whether the prior clinical data of good or bad quality. We also validated this important feature using COVID-19 and Alzheimer's disease, because for COVID-19 is a rare example in the sense that that's a new disease with no prior clinical data. So we built the COVID-19 disease model as early as March 2020, also published it. So by that time, and this is very challenging because there were no clinical data, very few animal data either in March 2020, just 2 weeks after the first case confirmed in China. But still, we can directly map the clinical phenotype of COVID-19 patients to the corresponding underlying mechanisms in our virtual human platform. And so we build this model in a totally zero data manner. But still, we use this model to publish predictions of outcomes of 52 COVID-19 therapeutics and vaccines of different kinds, and in a prospective manner, and achieve nearly 85% accuracy. So this case has sufficiently demonstrated that our virtual human platform, when it comes to evaluate clinical efficacy and toxicity of a novel medicine, it's just as if the medicine is tried in actual patient, but the interaction and the mechanism became relatively transparent to us in a still abstract manner. Also, we test our platform in Alzheimer's disease, which is a very different challenge. It's a complex disease with lots of prior clinical data, but very few prior clinical success. But still, we have also made multiple positive hits in the past. So both of these cases confirmed that we can make clinical efficacy toxicity predictions regardless of prior clinical data availability. We can go even earlier because if we know just the target and we can leverage existing clinical trial protocols, or and other hypothetical designs to evaluate whether a target could achieve good clinical efficacy and toxicity profile. So we also validated this capability using COVID-19 vaccine targets. So we, as you see that we predict as early as March 17, 2020, that all vaccines targeting S proteins only provide temporary protection at the expense of accelerating adaptive mutation. So this was made when there was no data to back this up back then, because that was the time when Moderna just injected the first mRNA vaccine to the first human volunteer. But now this prediction about the target has been fully validated. And when it comes to toxicity, we also had a very strong validation when it comes to metformin, which is the old small molecule drug for type 2 diabetes, but which has been widely regarded as a promising longevity medicine candidate for preventing the onset of age-related diseases. And using our platform, we have also evaluate metformin in the case of longevity medicine, basically, which means that metformin should be dosed to healthy populations in order to evaluate the primary endpoints like the incidence of age-related diseases like cancer, dementia, and so on. So we came to the very contrarian conclusion that metformin would have no genuine anti-aging effect. And more importantly, it's highly toxic, but its toxicity profile is a long-term cumulative toxicity. It's not acute toxicity for healthy population to take. So this is in contrast to the lots of data out there and lots of beliefs in the community. And our prediction has also been validated by the late published phase 3 clinical trial outcome, evaluating whether metformin versus placebo could bring clinical benefits to patients without diabetes history. And it shows that the metformin group has slightly higher incidence of cancer progression and significantly higher grade 3 toxic events. So this evaluation validation is very challenging. It's a twofold challenge, because first, metformin is a really complex, has very complex MOA, many different targets. On the other hand, this is in the context, the clinical setting of longevity medicine, which is more difficult than in the clinical setting for like a cancer treatment, because healthy populations, they have lower tolerability of toxicity. So thus we have completely multi-dimensional and the most rigorous prospective validation of the whole platform, such as currently our platform now enables very early as a target stage and highly accurate with about 79% of probability being right when we predict that a novel medicine could achieve good clinical efficacy and good clinical toxicity profile, even for first-in-class drugs in any indication. And so our platform can be leveraged, as you can see, at a different stage in the entire drug discovery and clinical development life cycle. And so that's all my introduction. I'm happy to take questions.
Yeah, thank you, Bragi. Very interesting. Any questions from the audience? So I wonder, how do you actually explain that because the algorithm, according to the numbers, seems to work, or platform, I should say, not only algorithm, seems to work quite well also in first-in-class situations, right?
Yes.
How do you explain that? Because first-in-class, by definition, means it's novel, right? So which means it's usually not on the data you learn. So how would you explain that? I mean, because that is, I mean, it's great if it works, right? But that is, at least, it's quite an extrapolation usually from the data you have, right? So how come, I mean, how would you rationalize that?
Yeah, that's a great question. So that it's best to come back to this methodological part. So basically, as you said right in the review, but directly just relying on bottom-up data-driven approach is not quite effective. So that's the reason why we need to first and foremost come up with a first principle-driven theoretical framework in order to generate a large number of high-quality hypotheses and as labels. So these, which means that these labels, they are not learned using currently quite limited machine learning algorithm, even reinforcement learning framework and graph convolution network. So these are not sufficient for generating the quantity and quality of the hypothesis. So put it another way, like aerodynamics, the humans we have been studying birds for thousands of years, collect lots of data about the different aspects that may contribute to the flying of the birds. But in the end, and when the human develop aircrafts, we first and foremost to have understanding of what is called the Bernoulli principle, which is a theoretical framework. So that's exactly the approach that we took, which we see as a hypothesis-driven approach to first come up with some framework, theoretical framework, which is analogous of a Bernoulli principle. Then we use it to generate labels. So that is the key difference.
Yeah. No, I understand that part. The question is though, in thermodynamics, right, you have physical systems probably generally are easier to describe. You can have differential equations or so, right?
Yes.
And often in biology, that it doesn't work as well because you go into stochastic limits, right? You have very heterogeneous systems. Cancer, let's say, the very heterogeneous types of cells. And you have usually here on the left, you have those 7 levels, right? From genes to species. Usually you have very conditional data, very noisy data, and so on. So in which way do you model the data then differently? Or the hypothesis, in which way develop better hypotheses than so many other people before, basically? Is there anything you can share about the key ideas or so? I mean, how will you do that in more detail?
Yes, so we definitely start from the least variant part of the data, which is that the genetic code. So there are only 4 standard nucleic bases, about 20 standard amino acids that is shared by different species across the entire phylogenetic tree. So first and foremost, we need to come up with basically the hypothesis for these really basic biological building blocks. Then we start and we build them layer by layer up. So that's why that we are not biased by the limits or availability of data generating techniques. So we start from these first principle building blocks with least variance, and then use it as a guidance towards understanding the data with really high noise and heterogeneity, and filling the gaps, and also to dealing with the potential conflicts or inconsistencies between data and generated in different ways.
Mm-hmm. Yeah, because in principle, I can completely follow the idea of having usually you don't have enough data. Basically, hypothesis space is too large, right? You have too little data, so you need to combine knowledge with data. And I think in many areas, to be honest, that will, oh yeah, it's basically what you describe here. I mean, I fully believe that. Yeah, the question is basically in practice how you do that. Yeah, looking forward to reading a paper when it's out, also to basically understand that in more detail. Yeah, how that's precisely done. Yeah, because that's key. Yeah.
Yes.
Okay. Any other comments or questions from anyone? Yeah, so the could you comment on you had that validation, right? So that was a prospective validation, you said, right? You were doing over 2 years or 3 years, yeah?
Yes.
Mm-hmm.
So the validation was actually carefully developed by our team in collaboration with stakeholders in the whole field. And because as everyone knows, we started by thinking about should we use our platform to develop a novel medicine like a disease-modifying first-in-class medicine for Alzheimer's disease. So that was the idea that was entertained by not only by us, but also by a number of the companies. But then we come up with, we also talked with other like healthcare investors, and they say that the statistical power is a key. And one trial, one drug may not really show whether your platform is good enough or not. You may just get lucky. And 10 trials, not enough. 20 trials, still not enough, but you need to reach statistical power. That's a very important criteria. So that actually motivated us that in order to demonstrate the capability, predictive capability in real world settings, we need to be able to not limit ourselves to specific indications. So that's actually what so it's a brainchild of the multi-party and collaboration to come up with this clinical prediction as the best way to benchmark the performance. And also we took it also to quite like highest level of rigor in order to establish like a standard, like a CASP challenge for protein structure prediction. So that's the reason why that we standardize the entire approach and using Twitter as a public venue, and in order to make sure that the entire validation has been realistic and informative from the application point of view, and measure the application setting as much as possible. And so this is not only our own idea, as you said, there are a few other companies in the AI-aided drug discovery field and adopted this approach, but their platform will build still heavily rely on deep neural networks or black box based approach. So they must use historical clinical trials to train their platform, but that actually impose a strong limit on their performance when it comes to first-in-class medicine. So that could partially explain the reason why that we could conduct the predictions in a highly comprehensive manner with high accuracy, whereas just using deep neural networks in a hypothesis-free approach can only achieve this performance at best so far.
Yeah. Okay. Good. Of course, the exciting thing then comes when you really use it completely prospectively, right? So in real drug discovery context, right? And you basically show with a pharma company, well, we increased our success rates to 85% or whatever the numbers is, right? Yeah. Yeah. Okay, very good. Excellent. Thanks a lot, Bragi. And we have two remaining presentations about read across. So the first presentation will be more on the fundamentals, and the second one will then be on particular applications and implementations of that. And I think Kathy is presenting first, if I'm not mistaken. Yeah, Kathy Lester from Procter & Gamble will present the fundamentals of read across. Yeah. Absolutely. Very good. You can see the slide.
Okay, very good. Let me. And let me get a pointer. Okay, good afternoon, everybody. I'm Kathy Lester. I'm a group scientist at the Procter & Gamble company, And today I'm going to tell you about our research in read-across and mainly quantitative assessment of similarity for read-across. And we all know that read-across is where data for a data-rich analog is read across to cover a data gap in a data-poor target or structure of interest. And its read-across is based on the premise that we can infer the toxicity of a compound based on the toxicity of a related compound. Now, read-across is, it sounds very simple, doesn't it? It's simple in theory, but it can be complicated and challenging in practice. And it is one of the important risk assessment tools we use at Procter & Gamble. We rely heavily on read-across. Back in 2010, my colleagues Sende Wu and Karen Blackburn developed a framework for selecting analogs for read-across and justifying their use. The framework is widely cited, and in 2011, they tested the framework against 14 case study chemicals, and the framework tested very favorably against those chemicals that possess toxicological data. The technique relies heavily on chemistry expert judgment, and it requires a chemist to demonstrate similarity in structure, metabolism, reactivity, and physicochemical properties to assign a rating of suitable, suitable with interpretation, suitable with precondition, or not suitable for a proposed analog. Suitable analogs are similar in all 4 attributes. A suitable with interpretation analog would be similar in the 4 attributes, but might be missing in one of them, or might be slightly less similar, maybe different phys-chem properties or something that would have no effect on relative toxicity. Suitable with precondition analogs usually converge metabolically. The analog and the target would converge metabolically, and not suitable analogs are missing at least 1 or 2 of these attributes, similarity in these attributes. Because the framework relies so heavily on expert judgment, it can be difficult for others to implement. It can lead to inconsistencies among chemists. It's not always transparent. So one way that we found to reduce some of these inconsistencies and the hindrance of transparency was to establish some rules for different classes of compounds. And we published these rules back in 2018. So the research I'm going to tell you about today is how some new tools that we've developed to reduce even further our reliance on expert judgment. So as Jennifer nicely showed, we know that structural similarity isn't always a good indicator of toxicity, and it's not a good indicator of analog suitability. And this slide pretty much drives that point home. What we did here, the P&G framework has been in use for since 2010, for 12 years. We have thousands and thousands of compounds that have been assessed, and we have like thousands of SOI analog pairs of compounds. So what we did here is we took about 3,300 SOI analog pairs of structures and computed what a similarity score would be. And that was just a Tanimoto comparison of the MDL 960 fingerprints, which are the fingerprints that are proprietary to BioVia. And we showed similarity scores, a function of molecular weight, for both not suitable analogs shown in red and suitable with interpretation analogs shown in blue. So we were surprised, very surprised by this, to show that there's just no consistency between similarity score and analog suitability. And what's an even more important point to be aware of is that a lot of tools for read across will use structural similarity for selecting analogs. And usually to do that, you specify a threshold, and then you will pull back structures with similarity scores above that threshold. And so you're actually missing out on a lot of suitable analogs that have lower similarity scores that we really can't afford to do in today's environment of limited toxicological data. What we did find for these 4,000 SOI analog pairs, that they were actually matched, could be characterized as matched molecular pairs. So matched molecular pairs it's a tool that's used heavily in medicinal chemistry. And it's a matched molecular pair of two molecules that differ by a defined molecular transformation, usually at a single site or small portion of the molecule. And I'm sure folks at this workshop are familiar with the technique. So, what a medicinal chemist does is he relies on these small changes in structure to increase his chances of increasing the lead, a hit for a lead for in medicinal chemistry. So he'd associate the transformations with a desired property like activity or log P. So what we've associated these small changes in structure, small transformations, is with suitability for read across. So you can see that if we have benzoic acid for a compound of interest, a methyl derivative would be considered a suitable analog. But we know when we have a bulky substituent in the para position of benzoic acid, there's associated toxicity with that. So that would be a not suitable analog. So when we analyzed a large number of SOI analog pairs for which the analogs were considered suitable, using our expert judgment-based read-across framework, we found that all of the matched molecular pairs, the transformations that we observed, fell into just five different categories for both aliphatic and aromatic compounds. For aliphatic compounds, which was about 30% of the compounds in the database, we saw some changes, mainly differences in chain length, which seem pretty obvious. Differences in alkyl branching of our chain, maybe some differences in bond order. The addition of a functional group was a small functional group, or if the structure of interest is a polymer or has repeat units, an analog with differences in repeat units might be considered. For aromatic compounds, those 5 transformations are a little different. We have some similarities. If we have an alkyl substituent on that organic molecule, it could be differences in the chain length. The addition of an alkyl group, the presence of branching, the relative position of 2 substituents on an aromatic ring, or even the presence of a functional group, and that would depend on what our scaffold is and the functional group. So in order to facilitate implementation of this non-structural similarity-based way to find analogs, mainly more substructure-based method, we summarized all of our transformations and what we learned from this exercise in a workflow. And we published this last year. So, So this workflow can be used to pull back analogs, right? You're going to find analogs, but to justify their use for read across, we have to show that we expect them to be similar in metabolism, physical chemical properties, and reactivity. So in the past, we chemists have done like a side-by-side comparison of metabolism. And so some of our new tools are quantifying that similarity. Can we quantify similarity in metabolism? So what we have done is we've defined a metabolism fingerprint. And what that fingerprint consists of are the biotransformations that are either known to occur for the structure of interest or the analogs, or that is predicted to occur based on data for similar molecules. So we chemists spend a lot of time when we have a structure that we're considering for read-across, we spend a lot of time learning about the metabolism. And what is known and what is expected. So these are two case studies that I'm showing for benzyl alcohol, and these are the analogs. And another one would be diisobutyl ketone, and these would be analogs. So the metabolism fingerprint for phase 1 or phase 2 biotransformations consists of those biotransformations. Those are the keys. And then reactive metabolism is just, that is the most important, one of the most important attributes to compare. Does the analog and the SOI, do they produce the same reactive metabolites? So we define a second metabolic fingerprint, which is reactive metabolite fingerprint. So that, and then list the reactive metabolism transformations known to occur, and then we'll specify the presence or absence of those transformations for the different molecules. For the diisobutyl ketone, we know that our 3-heptanone is going to be going to form. We know that will form gamma diketones. And we know our alpha beta unsaturated carbonyls also, right, are reactive, and our benzyl halides. So that's all taken into account. And to properly weight the formation of reactive metabolites, we take the product of the metabolism similarity score with the reactive metabolism similarity score to come up with a total metabolism similarity score that is shown down here. And we see when the reactive metabolism diverges for the analog versus the SOI, we get a 0 for the metabolic similarity. We do the same thing for reactivity. And in that case, the keys in our similarity fingerprint consist of alerts, and we use DARIC alerts, and we also use alerts from an internal DART assessment tool that was developed. So by combining the output from both of those tools, we can assess like the relative reactivity or an alert similarity. And again, our reactive, I don't know if I need to move this. Our reactive chemistries are reflected in those alerts as well. Finally, physicochemical properties are important, right, to demonstrate similarity and bioavailability. So what we did here for defining our fingerprint for physicochemical similarity was there are some papers written by Paul Gleason from GlaxoSmithKline, where what he did was he took the assays in their company and did some principal component analyses and found that these 3 properties were the most important. They could define more than 80% of the variability found in these ADME data that they had. And the properties were log-P, molecular weight, and charge, whether the compound was positive or negative. So we incorporated those properties into our physicochemical property fingerprint. And we also added volatility, because we know volatility is important. So whether or not the compound could be defined as a volatile organic carbon-containing molecule, so a VOC. So again, we did the same thing. You have the fingerprint, and we did use the Tanimoto comparison of those fingerprints to get a physicochemical similarity score. So finally, when we put all of these together. And if we just take the simple sum of all of these similarities, and our metabolism similarity, remember, is the product of metabolism similarity and reactive metabolism similarity, we actually come up with a total score that is sensitive to suitability for read across. So it is indicative of the suitability for read across. So what we did was we applied this approach to all 14 of those case study chemicals that Karen and Sunday published back in 2011. So this is that diisobutyl ketone again. And so on the x-axis, we have the total score as the sum of the different components versus a series of analogs proposed for this molecule. And you can see that the total score indeed does increase as we go from not suitable analogs to those that are considered suitable with interpretation, and to our suitable analog, we get the highest score. What we like about this approach, and this would be some data that we would hand off to our toxicologist, who would then actually do the read across from the data. They look at the tox data and read across to establish the safety of our structure of interest. So we see that this lets them rate analogs, like if they have more than one suitable with interpretation analog, this analog with the higher score might be a better analog, as well as they can see where's the outage for some of these not suitable analogs, like the analogs that form the reactive metabolites. We see that there's no, the metabolism similarity is 0 for those. For a compound with the longer alkyl chain, we see the phys-chem properties are off. The similarity with the SOI is much lower for that compound. So, so the diisobutyl ketone was one of those case studies. So what we did is we applied this to all 14 of the case studies that were published back in 2011. And found that indeed, for each case study, we see that for not suitable analogs, the total score is a lot lower than those considered suitable with interpretation, or suitable, well, not a lot lower for most case studies. And our computational toxicologist, Mahmoud Choubert, actually came up with a way to assess how well does this similarity total score distinguish between not suitable and our suitable analog. So how well can we distinguish these two? So what Mahmoud did was he looked at all different approaches. So he examined structural similarity just to make sure, are we missing any fingerprints that might be particularly sensitive to our analog ratings. So he looked at all of these different fingerprints, molecular fingerprints, to determine, can we use structural similarity to separate not suitable from suitable analogs? And you see that for none of these fingerprints can we distinguish the suitable from the not suitable for all 14 case study chemicals. We see that the MAX, and these are the open source MAX structural fingerprints, the fragments, I think there are 166 of those fragments. They seem to perform the best. It just might be the fact that the MAX fingerprints are just small structural features, and these are all small molecules. The SAGAR fingerprints were introduced from Siome, and those performed fairly well as well. But as you can see, structural similarity, we just can't use that, right, to distinguish not suitable from suitable analogs. So what Mahmoud did then was, what about each one of those similarity components that we have defined with our fingerprints and our Tanimoto comparisons? How well do those compare? And it turns out actually that the metabolism similarity works very well, right? We can distinguish suitable from not suitable. That's pretty important attribute. And then depending on the other, the chemical structures, we see that some of the other attributes are important as well. Case study 12 was a fatty alcohol, so something with just all the analogs had different chain lengths. So in that case, phys-chem similarity was the most important. But what Mahmoud did show that is if we take our total score and equally weight each one of these attributes, how does that compare? How is the separability that he defines? How is that? And we found that the equal weights works pretty well all across all 14 case studies. And then he did a logistic regression to determine what weights would give us the best separability, and found that with those machine learning weights of these different components, we could separate all 14 case studies using this approach. So where are we now, and what are our next steps? So for the matched molecular pair approach, for finding analogs, a structure-based approach rather than relying on structural similarity, like I said, we published that paper last year in Research Regulatory Toxin Pharmacology, and all of this QSIM work for our 14 case studies we are submitting to Chemical Research and Toxicity actually later on today. And then for looking at all of those thousands of SOI analog pairs that we have in our database, we are developing a metabolism database and populating that with the metabolism information that we chemists have compiled on all of the structures that we've considered for read across. And that's about 1,700 assessments we've written summarizing the metabolism of compounds. So we are building that database and populating that. And then at that point, we should be able to populate a database with the different similarity components for all 1,700 of our SOIs, and then however many thousands of SOI analog pairs we have. So we'll have some more data to show us what are the best weightings to use, and what is the best approach for coming up with a total score that is consistent with read across. So that's about it, and I just need to acknowledge all the people who do the work. Alante Bird is our expert in alert similarity. Mahmoud Shobair is our computational toxicologist. Gong Yan is a medicinal chemist who is an expert in metabolism. And Andrea Larson-Peters is our boss and helps us pay the bills. Other chemists that contributed to our read across approach are Mike Loffersweiler, Betty Niagodi, Ellison Rees, and Sende Wu. And I'd like to thank you for your attention, and I'm happy to take any questions.
Yeah, thanks, Kathy. And it's really, it goes back to some of the topics we discussed before, right? It's there's a degree of putting knowledge into the definition of your features, right? You have the metabolic similarity and so on, yeah? You don't want to
exactly
completely, otherwise you don't really know what the system comes up with, right? So there's a handcrafting, strong, in this case, strong handcrafting element in there, right?
Yeah.
That's right. Are there any questions from the audience? Yeah, there's also, by the way, I just put it into the chat, there's an NIH workshop on clustering and classification that actually touches on some of those topics again as well, right? So how do we actually use supervised methods versus unsupervised, right? So how do we weigh different types of features, what you showed on the last few slides, right? We'll visit that there as well. There's one, Elena has a comment. Elena, do you have a mic? And then we can discuss it orally.
Yes, thank you. So it is really a very nice work, and I find very interesting how to evaluate the similarity. It's not a specific question, but can you tell us or comment a little bit more about the metabolic similarity? just because I have seen these, well, not this approach, but the concept of metabolic similarity somewhere else, but it was never so well structured. And so I just would like to understand something more, please.
Yeah, you're right. It's not a novel concept. And I know there are some, there are some other implementations of this concept. I know Emilio Benfinati has established a metabolism similarity score. And I know Ovanis McKenyon, his group, they look at metabolism similarity and compare side-by-side comparisons of metabolite structure. And Emilio uses a similar approach where each, like a metabolism fingerprint is defined and each key is a biotransformation. And he uses the Sigma biotransformations that were published back in 2008. And actually that's where we started, but we found that we had to add to that compilation of the biotransformations. We had to think and create our whole new fingerprint for the reactive metabolite formation. So yeah, I think metabolism is probably the most important attribute for read across. Is something that we chemists spend most of our time trying to understand for each molecule that comes in. And unfortunately, that's the hard part, right, for others to implement if you don't have an army of chemists to worry about metabolism. Sometimes that's hard to think about.
Thank you.
Yeah, cheers. And there's one written question as well from Svava, and she asked me to read that out. Great work, first of all. And then to what degree will these methods be available to people outside Procter & Gamble? So are they publicly available as well?
That is something our group is working on. We're working on making these tools available, coding the matched molecular pair and making that available in like GitHub or something like that. We're publishing this work right now. And like I said, our metabolism is the most labor intensive, and we're working on our metabolism database and populating that. And I'm not sure our plans, I mean, working on our own predictor or something like that, our plans to share those are yet to be decided, but that is something to consider.
That's great. Kevin has a comment as well. Kevin, do you have a mic, or should I read that as well? Oh yeah, good. They asked me to read that. The question is, would structural similarity still be the primary screening feature for identifying potential analogs before calculating the total similarity score to select the most suitable analog?
Honestly, we don't. We don't use structural similarity to pull back analogs. We might use it in parallel with our matched molecular pair approach, where we chemists outline a search strategy for our search specialist who will make the modifications to the structure that we have proposed that have been outlined here to pull back the structure, the potential analogs. And I think this is Alante Bird, the expert in alert similarity. She is our search specialist. She also will use structural similarity sometimes, but typically doesn't find any new analogs that we haven't outlined here. So honestly, we don't rely we don't rely on structural similarity very much at all.
I can understand. It's more about defining categories that mean something. Similarity can be misleading, right? If it's not, if the knowledge is not put into this, it's too generic sometimes. In other words,
right, and I think that's pretty well accepted. that structural similarity is insufficient, right, for justifying the use of a compound for read across. You need those biological and toxicological descriptors or information.
That's right. No, read across sounds easy, but it isn't in practice. Absolutely. Yeah, great.
Yeah.
Okay, excellent. Thanks a lot, Kathy, for your contribution, because we will actually continue with the same topic, and Elena will present on practical implementations and applications of read across. Great. Cheers, Kathy. I will hand over to Elena then. Yeah, feel free to share your screen, Elena, and to start the presentation as well.
Unmute. Okay.
Does it work? Yeah. Can you hear us?
I can hear you. Okay. Now I have everything to share. Also the screen.
Yeah, audio is good, so we can hear you. I think the screen is coming as well. Yeah.
Okay. Good. Thanks a lot. So thanks a lot, Andreas, above all for organizing this Silico toxicology conference. So that I really think it is a good way to just to keep our community together and to keep the discussion going on. And thanks a lot to all of you, because you're still online, you're still with us after such a long day. Now, for the ones of you who don't know me, I just wanted to give you a couple of information about who I am and what do I do, so that you understand why I choose the content of the presentation as it is. So my background is, in a nutshell, to use computers to generate and predict properties of molecules. And just only in the last, let's say, 15 years, my focus has been toxicological properties. And in the last 10 years, the generation of toxicological properties for risk assessment. And nowadays, what I do, I am a consultant and I run training courses for the use of computational toxicology for risk assessment. So my approach is very practical, and I build the content of the presentation trying to give you some basics about the read across and an overview of resources, including case studies, that you can use to support your read across analysis if you want to start with this field. Trying also to meet a need that always applies on education. So when we last met up at the BTS in April, I heard that coming up quite often. So this is why I decided to build the presentation this way. Now, we are all familiar with the concept of the read across, and we already said it is easy in practice, but it is a little bit more difficult to run it. We have seen just now the concept of similarity. Read across is based on the concept of similarity. The problem is that to assess the similarity is not that easy, and the similarity is not a monolithic concept, but it comes with different flavors. I use the same terminology that Kathy has used, or the same concept in assessing the similarity. I just added this information that the similarity is endpoint specific. So according to the property that you are trying to predict, you might weight the different similarities with a higher or lower weight, because it really depends on the endpoint that you want to predict. If you're predicting an in vitro endpoint or an in vivo endpoint, as an example, the metabolic similarity might play a less relevant role. I add also something that is something related to the biological similarity. that we have seen today when I think Jennifer, I don't remember the affiliation, when she showed us how she's using biological descriptors together with structural descriptors, trying to build better models. So biological similarity is using this type of information to assess the similarity. So the read-across is, as I said, and as we all say, it is breedy as a concept, and how to implement is a little bit more difficult. And if you look out there, there are a lot of different guidance that can help you in implement the methods. Now we have the guidance of the OECD or the ECA that, in my opinion, are written sometimes in a too legalist way. So it's sometimes it is really difficult to put what they ask you to do in practice. While this technical report from the Easy Talk is a practical way of summarizing what these two guidance say, but in a much more practical way. So the content of these three documents are more or less the same, but this one comes with the more practical applications and also with examples. So in the latest 12 years, different frameworks were developed to address different use and applications of read-across. And if you want to have an overview and a review of the different frameworks, this is a very good publication that describes the different frameworks together with the applications. And what you can find also in this publication, it is a description of a sort of general workflow that you can use to put in practice a read-across analysis to the search, from the search of analogs to the data gap filling. And this is with just some very minor adjustments. It is the workflow that I personally use. So let's see this workflow. It includes 7 steps. And the first one, it is the decision context that is related to the fact that you really have to define the aim of your prediction. This is true for any method that you're using to generate a data, and above all for a read-across. So decide what you want to do with your number, and then this application will determine the level of uncertainty that you can assess. And we will see how you can assess the uncertainty level of your prediction. Once you've done that, you will have your target, that is the chemical for which you want to predict, to make your prediction. Hopefully, you know a lot of things about your target and or your target substance, that is the structural identity, the presence of impurities as an example. So you have all the information that you need to identify which is the structure you want to work on. And then you can collect the data, even using some QSARs or profilers, so that you can analyze what it is known about your chemical and build the hypotheses that will guide you to identify analogs. So you can use the existing profilers for genotoxicity, skin sensitization. There are some profilers for repeated dose toxicity, for developmental toxicity that you can use. And you collect all the data that you have, and you build a hypothesis. You will use then this hypothesis to identify your analog. And once you've done that, you can evaluate your analogs. And this is where the similarity assessment comes into play. So you don't use just the structural similarity to identify analogs. You use an hypothesis that can be a mechanistic hypothesis, as an example, and then you evaluate your analogs, carrying out different similarity analysis. And then there you can understand which are the analogs among the ones that you identified that you want to use for your read-across. And then the rest it is easy. You fill the data gap, applying your read-across, and then you can assess the uncertainty of your prediction. And we will see the elements to assess the uncertainties. The analog identification and the analog evaluation here, it is it can be a recursive process, in the sense that during the evaluation of your analog, you might reevaluate the initial hypothesis, because you might find something that doesn't match what was your initial hypothesis. So you can go back, redefine the hypothesis, and go into this process again until you find analogs, so you evaluate them, and you're happy with what you find, and you find nothing that is against your initial hypothesis. So there are several tools. This is a list of the freely available ones that can address one or several steps of these workflows, of this workflow. But the ones that is, say, the most advanced in the development, it is the well-known OECD QSAR toolbox that is also well-known to be quite difficult to use. And it is just difficult because it implements a lot of knowledge. This is a tool that is co-owned and co-managed by the OECD and ECA. And I say that it is in the most advanced level of development because in comparison to the other ones, because it is able to address literally all the different steps. So you can carry out all the different steps in the toolbox. Only one note related to the toolbox. The type of hypothesis that you can build and analyze in the toolbox are mainly similarity and mechanistic similarity hypothesis. You can consider a little bit the metabolism, but it has some limits from that point of view. What you can't do at the moment, it is to use the biological similarity. There are some of the Toxicology data there, but there are no tools to use this data and to assess the biological similarity based on this data. So at the moment, only the biological similarity is something that you can't really do in the toolbox. And the other tool that is using the Toxicology data for read across it is Genera, that is the biological similarity tool that is implemented in the CompuTox dashboard of the EPA. So let's have a look now at the uncertainty assessment and how we can assess the uncertainty of a read across. So again, we have frameworks that can help us, because at the end, the read across, it is a case by case. So it is difficult to give you a method that can always work. So this publication here, it is a tentative to list and qualify the sources of uncertainty of the read across. And it is based on a series of case studies that were developed starting from the Serena project that was dedicated to the development of approaches alternative to the animal studies. They produced a series of case studies, and then other case studies were produced, and the analysis of these case studies will just produce this list of sources of uncertainties. So the first source of uncertainty are the data of the source compound. So the data that you're reading across. And so these data must be adequate for the use that you want to do. And of course, you can't expect to have a prediction that is better of the uncertainty of the original data. So this is actually to take into consideration and whatever data you're using, you have to assess the reliability of the data of the source compounds. And then we have two groups of uncertainty. One, it comes from the similarity between the target and the source compounds. And again, here we find the different flavor in similarities. And then there is the uncertainty that comes from your hypothesis. So do you have a strong hypothesis that has a mechanistic behind? Do you have additional data to support your mechanistic hypothesis? And so you can reduce the uncertainty this way. In the publication, you also find some examples on how you can describe and assess all these different elements. And what I find interesting it is that these elements here that came from these case studies, they are reflected in the assessment element of the read-across assessment framework. Now, for the ones of you who are not familiar with Hemmerich and the read-across, the RAF, the RAF is a document. It is a framework that was published by ECA, but it was developed for the ECA scientists themselves because they had to assess the reliability of different read-across studies, and they needed to have the same system to apply in assess this reliability, so that different scientists, so that you could reduce the subjectivity in this evaluation. And the RAF is organized in different scenarios, and each scenario has a list of assessment elements. And what I've done here is that I listed the assessment element of scenario 2, that is analog approach for a property that doesn't change. So it is the same between the target and the source compounds. So these are the assessment elements that are related to the argumentation of the read-across. These are the ones that can be correlated with the similarity analysis. And this is the assessment element that is related to the reliability of the data that you're reading across. So these two frameworks, they go well together and they are superimposed, they are aligned actually. Even if the aims were different and even if what they are based on is slightly different. Now let's come to the existing case studies that you can use to learn or to develop better the read-across that you're working on. My suggestion is to use the IATA case studies that are published. The IATA case study project was launched in 2015. And the idea it was to ask stakeholders or institute or authorities to develop case studies where an IATA approach was applied, and to submit these to the OECD and to the team of experts. And this team of experts review these case studies, and then they highlight which is the strongest aspect of the case study, the uncertainty. They learn which are the areas for further development of the guidance, so they learn from these. And above all, what it is, information that I find interesting, it is which are the possibility of use of the case study. So you can find there given a read across with this type of justification and this type of supporting information, which is the possible application for which this read across could be accepted. This is the list of the case studies from 2015 to 2020. And as you can see, a lot, they have the read across as the type of assessment and different endpoints that were addressed. So it's quite a rich source of information. And then if you're working for Roche and ECA themselves, they published this read across illustrative example. It's not very rich of information, but it is in any case an example. And the ones that are really interesting are the illustrative example with the OECD QSAR toolbox, so that they give you some example on how you can apply that tool in a proper way. The endpoints addressed by these examples are skin sensitization, short and long-term aquatic toxicity, and acute toxicity. Now, just to finish, what if your substance is a UVCB? So what if your substance is a complex mixture that you can't even completely define? Well, in this case, and ECA had to deal with a lot of UVCB. So ECA was the one who started to publish something on what to do if you have a UVCB. So you have two documents here. One is very recent in 2022. And the approach that you can apply is literally, or you use the constituent of the UVCB and the data that you have for the constituent to read across to the whole substance, or you use another substance that is similar to your target substance, and you read across the available information from this substance. There is not a lot, and there are not a lot of case studies related to UVCB. This is an example of one that has been published a few months ago, and I work with this group. It is an interesting case study, and I work with them to build with them what was the similarity assessment, and in this case we use a constituent-based approach. So we use a different constituents and we assess the similarity with chemoinformatics and some chemoinformatics methods. The last thing I would like to add related to the UVCB, it is that in the latest document from ECA, it's interesting that they are starting to be open to the possibility of using analytical fingerprints to analyze and assess the similarity between two substances. So there is also this possibility that was not included in the previous document, and I think it is a very good and important development. Other application of read across can be applied in different ways. One that I find good, it is to search and to select a PBPK model, reading across. So you find molecules for which a model has been developed that are similar to your molecule. And so in this way, you can find if there is a PBPK model that is already there, ready to be used with just a slight few adjustments. And they also put together, this group also put together a database of the existing PBPK model with the source compounds. And then there is this interesting approach that is using to use omics data to assess the similarity. And yeah, it is at the beginning. I know that they are further developing what it is included in this publication in a bigger project. And that's the last slide. And if you have any questions, I'm happy to answer.
Okay, now it works. Excellent. Thanks a lot, Elena, as well. Any comments or questions from anyone? Okay, so let's see if there's something on the. How do you? Yeah, I think Jose's question is actually, I think that's a new one from you. How do you handle activity cliffs and MMP cliffs in read across?
Well, it's I mean, so of course, if we are dealing with the one-to-one read across, you will not be able to see activity cliffs. If you are the idea is that you should find more than one analog, and in that case, should be able to see something. Of course, if we are talking about a case like the different stereochemistry, this is not something that is possible to assess with the read across. But fortunately, this is a rare case. And actually, usually the read across is very effective with the low toxicity chemicals. So they are not supposed to be biologically very active.
Hope that answered that question. Anything else? Any other questions from anyone? Okay, let's just check here. I think YouTube is fine. Okay, good. I think that actually is the end of this presentation as well as the event. Thanks a lot for staying around. Thanks a lot to Elena as well, much appreciated. Thank you for joining. I hope you had an interesting time. I will cut the recording as well, so the vast majority of the presentations will be available on YouTube afterwards. And yeah, please feel free to check the event website. Then many of the slides are on there as well as the presentations will be on there. Okay, great. Thanks a lot, everyone, for joining. Hope it was interesting and looking forward to seeing you again next time. Thanks a lot. Have a good evening. Bye-bye.